Great, it seems we are alive, so we can start today's work. Pass the word along to Diogo Figueiredo that will start today's session. Thank you, Diogo. Thank you, Thulani, for coming here, and Juliana for being here, so we can go ahead and jump right in. Thank you, Diogo.
I think we are live, right?
Yes.
Okay, so welcome everyone. We are here today for the first day of the Biotec Bioinformatic Tools and Applications Workshop. Before we get started with the workshop, I would just like to present myself. My name is Diogo Figueiredo. I'm a consultant at SPI, and we've been working together with Professor Fabian and Professor Krieger to organize this workshop. Just to give you a little bit of context, this workshop is one of many activities from the All-Atlantic Cooperation for Ocean Research and Innovation Project, which is called ANCHOR for short. It is a project funded by Horizon 2020 under the umbrella of the All-Atlantic Ocean Research Alliance, and its main goal is the implementation of the Belem Statement, with the aim to upscale research and innovation cooperation within the Atlantic Basin, also to better understand and sustainably manage the Atlantic Ocean as a whole, and to see the basis for healthy future large-scale joint Atlantic marine activities. Today's workshop is a part of the GPA, the Joint Pilot Action AA Biotec which is short for the All-Atlantic Marine Biotechnology Initiative. This joint pilot action of the ANCHOR project aims to foster collaborations across governments, across academic, private sectors, in the fields of marine biotechnology. It also aims to create mechanisms of technology transfer and promote the development of new marine biotechnology companies at the Atlantic Ocean level. I'll be here today to help Professor Krieger with the workshop. Any questions you have or anything you need, just tell us, and if we can be of help, we'll be sure to let you know and take care of it, of your questions, of anything you need. I'll also take this opportunity to inform you that the next event of the AA Biotech MAR Joint Pilot Action will take place on June 27th and June 28th, and will focus on the first day of this event will focus on aquaculture, and the second day will focus on a startup accelerator. So just to conclude, I'd like to thank you all for your participation, and I wish you an excellent event. I'm now going to pass the word to Juliana. Juliana, you can go ahead. Thank you very much.
Thank you, Diogo. Well, welcome everybody. It's a pleasure to have you all gathered here today, this morning. And we're gonna welcome now Dr. Thulani Makhalanyane. He is an associate professor in the Department of Biochemistry, Genetics, and Microbiology at the University of Pretoria. His scientific interests focus primarily on fundamental questions in the field of microbial ecology, genomics, and microbial metabolic activity. Understanding microbial community dynamics in natural systems is a central goal of his research. Thulani's current research spans across ecologically extreme terrestrial and marine environments. Dr. Thulani, welcome. It's a pleasure to have you here. So you have the word.
Thanks, Juliana, and thanks for the kind introduction. Hopefully everyone can hear me and you can see my screen. So I had initially wanted to talk about some of the work that we do in the Southern Ocean and also in Antarctica, but I decided to just stick to the Southern Ocean because it is quite a lot of work over a very diverse geographic range. I first want to thank Ricardo and the organizing committee for allowing me the opportunity to share my work. So over this lecture, I will try and give an introduction of the microbiome and then take you through some of the work that we do. Throughout the talk, I will discuss some of the computational approaches or the bioinformatic approaches that we use and highlight some of the key results that we've revealed by applying some of these tools. So I work in the Southern Ocean and also in Antarctica. So I will, in the middle of the talk, give you a little bit of an overview of the work that we do in Antarctica and why we want to focus on these areas. So this is our research group at the University of Pretoria. So we are dedicated essentially to understanding the diversity and function of microbial communities across a variety of different ecosystems. We are currently about 16 researchers in the group, so it is quite a big group. And we are led by myself and Oliver Muhasa, who's a senior bioinformatician who's working in our group. So one of the quotes that I always think about when I think about the microbiome is a quote by the former Secretary of Defense in the United States, who said, "There are no knowns. There are things that we know. We also know that there are things that are known unknowns." That is to say that we know there are things that we do not know, but there are also unknown unknowns. The things we don't know, we don't know. So if that quote is not confusing enough, I think it's just a perfect description of what we know about microbial communities in the environment, or briefly, just what we know about the microbiome. So maybe as a working definition, as we go through the talk, I'll give you the definition I'll be using to define microbiome. And that is that a microbiome is essentially a collection of microbial communities and how they evolve over a well-defined habitat. As you can imagine, these habitats are quite wide-ranging and diverse. They can include everything from host-specific microbiomes, everything from leaves to the air to deep-sea hydrothermal vents. So they are quite diverse and wide-ranging. Our work focuses on microbiomes in the Antarctic and also in marine environments, and I'll give you some of the reasons why we've chosen to focus on these environments and some of the underlying motivations for this work. So we do know that there are still, like the quote says, many unknowns regarding the microbiome. So we know essentially that these communities are more or less ubiquitous across all different ecosystems. We also know that these microbial communities are numerically very abundant. There's some estimates that say that in any environment you'll find essentially 1 billion different bacteria, right? So that's not counting the number of archaea that may also be present in that particular environment. We also know that about 99% of the organisms that are out there are essentially recalcitrant to the conventional methods that we use to culture microorganisms. So most of the organisms that are out there remain essentially unknown and uncharacterized by the current conventional methods. So it's important, of course, to know what these microorganisms are, because some of them, a small proportion, may lead to adverse health outcomes, or they may negatively impact ecosystem function. For example, we know that some bacteria are important allergens. Bacteria and fungi are important allergens, and that some of these allergens may account for millions of sick people in various countries. So not knowing what these organisms are could essentially have bad outcomes in terms of the negative health effects that may come from not understanding the diversity of the microorganisms that are out there. So we know that microorganisms are, as I mentioned earlier, ubiquitous across a range of different ecosystems. So we know that microorganisms also play very important roles in cycling important nutrient elements. So they're essential for cycling nitrogen, help plants fix the nitrogen that they require to carry out their ecosystem services. And we know that microorganisms also play an important role in sequestering carbon dioxide or atmospheric carbon dioxide, a lot of which is done or occurs in the oceans. What we actually don't know is what will happen, as an effect of climatic shifts within the environment. How will these climate change processes impact on the abilities that microorganisms have to carry out their important ecosystem functions? And that's in part what our studies aim to do. So what's shown there, it's a famous depiction of the tree of life, and that shows eukaryotes over there, with us and our nearest ancestors, the plants that are very close to us. Although a lot of this tree shows that a range of the organisms that are out there, either bacteria or archaea, what you can see is that only a small proportion of what's displayed in the tree of life is of eukaryotic origin, and the vast majority of the organisms that are out there are microbial. What the tree doesn't show is also, the concomitantly high level of viruses that are out there, that are a source of genetic diversity, and that may also impact on the diversity of organisms that are out there. So in the microbiome group, we've got about 4 different thematic areas. We are interested in understanding microbial processes, adaptations, and potentially applications. So we've got a program where we're doing some bioprospecting for potential novel enzymes. These may include things like stress response genes, and a lot of these are also being functionally characterized and analyzed as part of different PhD programs. We are interested in understanding the microbial ecology of extreme local and also host-associated microbiomes. So, and I'll talk a little bit about part of that work. We are interested in understanding geographically strategic marine environments, and this is where it links up to the work that we do linked to the signed Belem agreements, where we are looking at microbiomes in the South Atlantic in particular, but we've also got studies where we're looking at the Indian Ocean and also the Southern Ocean, which of course, due to the connectivity of marine environments, is related. And I'll talk a little bit about some of the results from the South Atlantic and also from the Southern Ocean. We've got a range of projects where we are looking at the gut microbiomes of South African individuals, and this work, includes everything from characterizing, everything from unknown crassphage viruses and how these can be used essentially as markers of ecosystem change and also contamination in pristine marine environments. So it's a big portfolio of different studies, but this is just a small overview of the work that we do. So the current projects that we have in the group can be characterized or classed into these 5 classes. We're looking at the microbial ecology of South African soils. We are looking at human gut microbiomes of urban and also rural individuals in South Africa. As part of the South African National Antarctic Program, we've got a program where we're looking at the ecology, evolution, and function of marine microbiomes in the South Atlantic and also in the Southern Ocean. We are part of a large European, Brazilian, and South African consortium, which is focused on understanding microbiomes in the South Atlantic. And we also have ongoing activities where we're looking at the microbial ecology of Antarctic permafrost ecosystems. So you might have noticed that I spend a lot of time talking about polar and marine environments. So as South Africa, we've got, of course, a strong interest and a geographic advantage in studying some of these systems. South Africa has about 3,000 kilometers of coastline. We are surrounded by the Indian Ocean, the Atlantic Ocean, and the Southern Ocean below us. And of course, that forms part of our interest in studying and looking at processes that occur in the Southern Ocean and in the oceans around us. We've got a base in Antarctica, and this forms part of part logistical cruises that go down to the Southern Ocean. In fact, at the moment, my team or part of our students have gone down to the Southern Ocean, where they've gone down to the South African base that's on Marion Island. So there is, of course, a very strong scientific justification. We know that oceans transport nutrients and heat, and they are important regulators of the Earth's climate. So previous studies have shown that the sequestration of the world's carbon dioxide can occur through two different mechanisms. The first being the solubility pump, where cold carbon dioxide containing water essentially sinks to the bottom of the ocean. The second can occur through what is known as a biological pump, where inorganic carbon dioxide may be converted and eventually sink into the bottom layers of the ocean. So the role of phytoplankton and zooplankton as drivers of these processes is increasingly very well characterized. But we lack the comparative understanding of the role that microorganisms, including bacteria, archaea, and fungi, and viruses play in regulating some of these processes. And we also don't know how these microbial communities may respond to the current perturbations or changes that are occurring in the oceans. We know as well that oceans are important and regulators of climate change, and that they transport heat across different geographic scales, in addition to their role as vessels or vectors of transporting goods and services across the world. The Southern Ocean is also particularly important due to its important role. It's a regulator of the Earth's climate and its connectivity with the Pacific, Atlantic, and Indian Ocean, and how it regulates the Earth's climate. So in South Africa, there is currently a very strong support for fundamental research on Antarctic ecosystems. We've contributed since inception to the Scientific Committee for Antarctic Research, SCAR, since 1958. And as part of the ongoing stewardship or role that we play, carrying out research in Antarctic systems is one of the key roles that we play as part of our contributions to SCAR. We are also involved in ongoing international efforts to essentially establish a global microbiome and biotechnology initiative. And this is also linked to ongoing activities or agreements, as mentioned earlier, through the Belem Agreement. So this is just a broad overview of why we are interested in understanding these environments. And I'll then talk a little bit about the work that we do in the Southern Ocean. So as I talked and introduced earlier, that our system is the Southern Ocean. It's an important marine environment in the sense that it surrounds Antarctica, and because of the unique environmental variables that are part of the Southern Ocean, it is a very key marine environment. It is also the strongest ecosystem in terms of some of the currents that are embedded or part of the Southern Ocean. The Antarctic Circumpolar Current is also an important regulator or vessel of transporting heat and nutrients across the world's oceans. The Southern Ocean is vital in terms of its role for marine and global environments, as I've mentioned, as due to its role in transporting nutrients and heat across the Earth's climate. So we know that the Southern Ocean is also a major carbon and carbon sequestration source, and also it plays a major role in terms of its ability to store and transport heat. Roughly 40% of anthropogenic carbon dioxide from the atmosphere, it sinks in or is absorbed by the Southern Ocean. We know that 75% or so of the total ocean heat uptake is ascribed or linked to the Southern Ocean. We know that phytoplankton and also prokaryotic communities are important in driving both of these processes. microbes are important in sequestering carbon dioxide. They play an important role in the biogeochemical cycling, carrying out important processes such as nitrogen fixation, which occurs, of course, in the oceans. They also play an important role in sustaining local and global food webs. Phytoplankton and microbial community actions are often the basal stage of oceanic food webs. So it's quite important to understand the diversity and composition of the microbial communities that are in the ocean. The Southern Ocean, as part of its unique traits, is also an interesting environment due to its known high nutrient, low chlorophyll regions that are intermittently dispersed throughout these oceans. So the fact of these varying nutrient conditions in the Southern Ocean makes it a very interesting ecosystem, because the limitation, for instance, of certain nutrient variables such as iron is potentially an important regulator in the ability of microbial communities to carry out important ecosystem services. And not a lot is known regarding how, for instance, trace element limitation impacts on the ability of microbiota in the ocean to carry out the important ecosystem services. So as part of this study, I'll talk a little bit about the research that we did in the past 2 years, where we took 2 cruisers down to Antarctica along this transect aboard the RV Agulhas 2, and we looked at the microbiota in the Southern Ocean in winter and spring and asked lots of different questions. But one of these was the impact of how a spring bloom impacts on the diversity and structure of microbial communities in the Southern Ocean. So I'll give an overview of the typical methods that we use in the lab. The first, of course, we start by sampling, and this then follows by a combination of different approaches. One of the things that we do very routinely is, to get an idea of the quantity or to quantify microbial communities is we undertake a flow cytometric analysis in all the samples that we do. And that, of course, gives us an ability to estimate the abundance of microbial communities. We also carry out either a combination of 16S rRNA gene approaches to look at the diversity and composition of bacteria and archaea in marine environments. And these are typically sequenced through an Illumina platform. And we usually select either the V3 or V4 regions as our choice markers. And at times, it depends on the precise questions. We could have primers or markers that are specific for bacteria, or some that target bacteria or archaea, or some that have a combination, depending on the type of questions that we want to ask. We normally then do a combination of different amplicon analysis. We typically use a lot of CHIME2 analysis, but increasingly we are using the Data2 platform in terms of generating amplicon sequence variants, and then these are always compared to existing databases in the Silver database and so on. And then we also do a lot of shotgun metagenomic analysis. And as part of this, because of the differences in the costs associated, we'll typically, in any kind of project, take a subset of the total samples that we would have used for amplicon analysis. Once we obtain some information on the structural variation, a subset of these samples would then be put through shotgun sequencing, where we then would ask other questions related to the function of these microbial communities. Of late, we've also then used the shotgun metagenomic data to do things like genome-resolved metagenomics, and I'll talk about a little bit of some of those insights later on when I take you through the talk. But a lot of this then would do classically the metagenomic assembly, reconstruction of metagenome assembled genomes, and also then do some functional annotation, depending on the type of questions that we have. So this is the typical bioinformatic workflow that we'll do in the lab. And I'll give you two examples where we've either used the amplicon analysis approach to ask different questions regarding microbiomes in the Southern Ocean, and then talk to you about some of the genomic insights that we've learned through the shotgun sequencing approach. So the first question that we were interested in understanding and asking was, what effect does a spring bloom in the Southern Ocean have on the abundance and structure of microbial communities? So for this, of course, we followed the amplicon sequence analysis, and I'll give you an overview of some of the key results. So we took samples in winter and spring and followed more or less the same transect. And what we did here, we looked at microbes all the way from euphotic zones of the ocean, the first 5 centimeters, and all the way down to about 10 meters above the sea surface. So we did a whole depth transects throughout each of these different points. We wanted to understand what the effect of a spring bloom, which occurs in early spring in the Southern Ocean, how that would impact on the diversity and composition of microbial communities in the Southern Ocean. What we found is that the shift that coincides with the spring bloom led to increases in things like the chlorophyll A concentrations, which implied that prokaryotic growth in the upper chambers of the ocean may be supported by phytoplankton, photosynthetically derived dissolved organic matter. And this is also a finding which is more or less consistent with what has been found in other marine environments. So this consistency in the prokaryotic cell counts, as you can see here, seems to suggest that there may be more or less a fast remineralization of organic compounds in apologies, my office phone has decided to ring. I'm just going to stop it. Okay, sorry about that. I forgot to unplug the phone. And so I was talking about the consistency of prokaryotic counts and how this may suggest a fast remineralization of organic compounds in the epilegic regions of the ocean, and a slow sinking of organic matter, which then tends to support the elevated heterotrophic growth that we saw below 500 meters in spring in our sample sets. We also observed that prokaryotic cell counts were higher in spring in the epipelagic zones, and that we also showed no differences between the seasons in the aphotic zones. So this more or less resilience of microbial communities moving from winter to spring, as I mentioned earlier, may suggest that there's a rapid remineralization of organic compounds that occurs in the surface of the oceans. We also noticed that there was a decreased abundance in higher latitudes, that is in the first 500 meters of the oceans, as you can see from this figure here, but primarily more in the higher latitudes. So we also showed evidence that there was very limited compositional and diversity variation with the onset of an early spring bloom in the Southern Ocean. So our data analysis showed that there was limited taxonomic variation between the seasons. We, in fact, showed very little, at least at phylum level, we observed very little changes moving from winter to spring, even though there was that spring bloom that occurred. But we did see some changes in certain taxa. For instance, cyanobacteria were much higher in spring in the epipelagic zones of the ocean compared to winter. We also showed that there was no increase in taxa that are known to be typical bloom responders. This is based on findings from other environments. Here we saw, for instance, that Florobacteria and other gamma proteobacteria did not increase as we expected in the Southern Ocean. We also observed no increase in archaeal communities in spring, and this was also in contrast to what has been observed in other marine environment. So there are various reasons for this lack of responsiveness in terms of the microbiome. It is possible that the very low nutrient conditions in the Southern Ocean may impact or pose limitation in terms of microbial communities' abilities to rapidly respond to changing environmental conditions. Of course, the temperature in the Southern Ocean is much lower than those from those in previous studies which have observed some of these variations. So temperature may also be posing a strong metabolic barrier, a limitation, limiting the type of responses that we would have observed. We also saw that alpha and beta diversity did not differ as a response to the bloom. However, depth and latitude also seem to be determined by the changes in phylogenetic diversity. Bacteria and alpha diversity was significantly lower in the epipelagic zones of the oceans compared to the higher latitudes. We also observed that the community structure seems to be, in our study, more influenced by latitude and water mass endemicity in both bacteria and archaea. And you can see some of the results as displayed in these PCA ordination plots. I'm not sure whether the plots are big enough for you to see, but you can see based on the different colors that the microbiota that are linked to specific depth tended to cluster together. So it shows discrete niche specificity in terms of the different depths going through the oceans. However, when we looked at the trends of microbial communities by comparing both the data sets from winter and spring, as you can see from this mixed assortment of colors, there was really no differences regarding the microbiota in spring and winter. But the patterns seem to be more explained by the differences in depth rather than the seasonality or even the effect of the spring bloom. So when we try to understand how different physical-chemical variables may explain the patterns that we observed in terms of microbial community structure, we found that, interestingly enough, that at the higher depth of the ocean, in the first 50 meters, you see a lot of variables such as the ammonium levels within the winter component of our sample set seems to be a much more substantial driver explaining community variability, and that when you looked at the same data sets or the same points in winter, it seems like there is also a consistency in the sense that in addition to the ammonium levels, you see things like nitrate and urea, and also the chlorophyll A concentrations, explaining a lot more some of the variability that you observed in the upper layers of the ocean. So there could be lots of reasons for that, and I'll put on the slide essentially two different hypothesis. We know, of course, that microbial communities are known to be structured by depth in the water column, and this is more or less confirmed by the results that we've shown, and this is irrespective of the season. There could also be, it is also possible that the organic matter that's exported in the Southern Ocean may be insufficient to support discrete differentiation in terms of specific niches. Another possibility is that when we sampled during an early spring bloom, it may not be necessarily a true representation of what happens at the later stages once the bloom has set in the Southern Ocean. So what we also tried to do, and I'm conscious of time as well, I'll talk a little bit about some of the data that we tried to, that we observed when we looked at the co-occurrences of different bacteria and archaea in the Southern Ocean. So the results that you can see here in terms of what we found when we looked at different co-occurrences in winter and spring is that you see a diverse assemblage of taxa in winter, while the different hubs in spring seem to all be ascribed to alpha proteobacteria. We also observed that there might be important putative heterotrophic SAR11 bacteria that are known to, for instance, consume phytoplankton-derived organic matter that may be important bacterial keystone taxa, looking at the data set that we observed, that we collected from both winter and spring. When we looked at determining potential keystone taxa for archaea, what you observe here is that there is essentially much less diversity in terms of the hubs that you observed within taxa. The network hubs that we showed here are essentially what we saw in the deeper layers of the ocean, where it seems that archaea seem to play a much more substantial role in driving connectivity among different taxa in the Southern Ocean. We also observed that certain taxa, for instance, seem to be much more common in the spring compared to the different network hubs in the winter data sets. So this result shows that, in contrast to what we observed earlier, where it looked as though the diversity of organisms was much more consistent between winter and spring, is that you might see the emergence of specific keystone taxa that drive certain processes when you look down at the co-occurrence patterns of these taxa between winter and spring. So maybe just to summarize part of these results, is that what you see is that there is a smaller effect of phytoplankton bloom on microbial communities than we would initially have expected. And there are different reasons for this, which I've outlined separately, but I'll mention that there was, for instance, an increase in prokaryotic cell numbers, and that cyanobacterial relative abundances seem to have been higher, and that the bloom did not seem to have an effect on aphotic zones of the oceans. When you go down, when you look at the deeper levels of the ocean, you see much more resilience in terms of the community structure. The diversity and community structure was also not affected by an early spring bloom, as we initially had hypothesized. And there are, of course, again, lots of different reasons or possibilities for this, and I've outlined at least 5 of them here. There might be due to a resilience of the microbiota in these environments. It could be as an effect of the, as I mentioned, the trace element limitations that occurs in the Southern Ocean. We know, of course, that iron is very important in terms of driving productivity of microbial communities. And the known limitation of iron in the Southern Ocean may explain some of the resilience or the lack of variability in microbial communities that we observed. And there could also be an alternative explanation to this, is that there might not have been sufficient time to observe the full effect of a spring bloom in the Southern Ocean, because we sample more or less in the early period of the spring bloom. So the second example, which I'll quickly go through, is how we used genome-resolved metagenomics to understand specifically chemolitho-autotrophic bacteria in the Southern Ocean. So here we essentially asked whether the Southern Ocean microbiota have potential to fix inorganic carbon dioxide in the dark ocean. This was motivated, as you would have observed in the previous result that I talked about, where there was more or less a resilience in terms of the microbial communities that were observed in the aquatic zones of the ocean. So briefly, just to go over what we did in using metagenome assembled genomes, we constructed about over 600 mags from the Southern Ocean dataset. And what we saw is that about 216 of these mags are potentially novel, just based on things like average nucleotide identity and other different matrices. This suggests a high degree of endemism in the type of microbial communities that are observed in the Southern Ocean. When we look at some of the patterns, for instance, the genome size, the GC content and coding density in each of the taxonomic groups, you see a discrete differentiation depending on the type of bacteria that you are looking at. In other words, if you look at genome sizes, for instance, there is a lot of similarities in, as you would expect, with microbes, in particular different phyla. One of the interesting things that we observed is that, in contrast to Southern Ocean prokaryotic communities, the genomes that we observed here had very comparatively large genomes, and that the GC content also seems to be comparatively very high compared to other marine taxa. For instance, if you look at the gamma proteobacteria and SAW 3T4 in our data sets, they seem to have much higher genome sizes and higher GC contents compared to other organisms that are out there. So this is, of course, ongoing work, but what I'm showing here is that there is a really high diversity of metagenome assembled genomes that we observed in the aphotic zones of the oceans, and a high proportion of this, about 114 mags, seem to be affiliated to Archaea, and a high proportion of these, as I mentioned previously, seem to be unique to the Southern Ocean, just based on the fact that they have very low similarities to other comparative genomes from other marine environments. 167 of these mags also possessed unique metabolic traits linked to the ability or the capacity to cycle inorganic carbon dioxide pathways in the Southern Ocean. And this is an interesting trait given what we observed in terms of the functional dynamics, especially as you look down to the water lower columns. So when we looked at characterizing the metabolic traits of chemolitho-autotrophic bacteria, we can see, as you can see from this plot here, that the genomes seem to be metabolically diverse. The chemolitho-autotrophic taxa that we observed in our data set seem to possess the ability for thiosulfate oxidation, ammonia oxidation, nitrate oxidation, and also some traits linked to methanogenesis. So most of these organisms appear to rely on the Calvin cycle or the reverse TCA cycles as a primary means for obtaining inorganic carbon from these. And a lot of the organisms that we observed, or the genomes that we observed, seem to also have a diverse array of reduced inorganic nutrients that are used as more or less as an energy source by these organisms. So a lot of the genomes that we also observed, though we won't go into details, seem to also show evidence of following mixotrophic metabolisms or lifestyles in the Southern Ocean. Maybe just to briefly summarize the results. So what I've tried to take you through is how we've used a combination of amplicon-based analysis and also shotgun metagenomic analysis to ask different sets of questions regarding the diversity of microbial communities that are in the Southern Ocean. In the second part of the talk, our questions were essentially whether Southern Ocean microbiota have the potential to fix inorganic carbon dioxide in the dark ocean. So interestingly enough, we recovered over 200 potentially novel genomes, which we believe is the highest compendium data set of novel Southern Ocean specific genomes. A lot of the bacteria and archaea that we observed harbor diverse metabolic traits linked to the ability to fix inorganic carbon through what are primarily chemolithoautotrophic means. We also observed that at least in the aphoric zones, chemolithoautotrophy seems to be very widespread in the Southern Ocean, and also it's irrespective of specifically which geographic or latitudinal station that we sampled in our data set. We observed that there might be selective pressures in the Southern Ocean, as I've talked about, the trace element limitation and the other physical chemical constraints on microbial communities that may induce a high degree of horizontal gene transfer events as observed in terms of the results I didn't talk about here from our pangenomic analysis, where we looked at chemolithoautotrophic genes between different prokaryotic domains, that is between different bacteria and archaea in the Southern Ocean. So there are several different ongoing questions also linked to our work in the Southern Ocean. We are interested in understanding further how the interactions of microbiomes or microbiota in the Southern Ocean could potentially act as a feedback or influence biogeochemical cycling in the Southern Ocean. So to do this, we have got several ongoing studies where we're looking at nutrient co-limitation and its impact on structuring bacterial diversity, and also in terms of measured function using things like extracellular enzymatic assays to assess different capacities in the Southern Ocean. We've got ongoing projects, I didn't talk about the viruses a lot here, where we're looking at viral host interactions and potentially how these may also be important drivers of phylogenetic diversity in the Southern Ocean. We've got several projects where we're also looking at understanding how community level processes influence diversity in the Southern Ocean. We're also interested in understanding how adaptations to climate change may impact the diversity of organisms, but how in turn these organisms may act as potential feedbacks, further exacerbating the effects of climatic shifts. With that, I'll stop here and take questions if there are any. Thanks a lot for listening.
Hi, Thulani. I think you are back. Are you listening? Are you able to hear me? So the members of the room can return, make us company, but we have several questions coming from YouTube, and we can, if you can entertain us by answering those. Uh, the first one is from Jessica. She's asking, since we study omics to understand the web dynamics of microbial marine community, can you use this data to infer the dynamics of the benthic community due to the presence or lack of nutrients?
So one of the things I didn't mention is that, of course, one of the challenges that we had is that classically oceanographers tend to look at nutrient dynamics only in the euphotic zones of the ocean, and we lack, for instance, data on the nitrite levels, going down the water column. So, and we also know that microbial communities change, right, depending on the effect of nutrient variables. So that means we cannot directly make inferences on observations that we see on the surface in terms of what would happen going down the water column, because the factors that shape and drive the microbial communities, as you mentioned, for instance, in the benthic communities, may be very different from those that occur in the surfaces of the ocean. So the only way really to make those inferences and to study those dynamics is by collecting microbiome data sets from other deeper layers of the ocean in order to be able to study the patterns of microbial communities.
Okay. I have a question on my own. Can you see already changes in microbial communities due to the anthropogenic actions? Like, for example, high salinity or low salinity, high acidity or low acidity on your samples, and especially the temperature that you are, of course, you are monitoring, month by month, year by year. Can you see that already happening in the South Atlantic? That's something that's already striking.
Yeah, so we do observe that. I mean, just as a testament of looking at microbes in different water masses, you already see, because different water masses will be defined based on their salinity contents and so on. You'll see a discrete structuring of microbial communities based on these variables. And of course, temperature is a substantial driver because it impacts on the metabolic rates of microbial communities. So that impacts on the structure of microbial communities very clearly. We've also done other studies where, because it's of course not easy to look at these interactions by studying microbes in the environment. So we've asked other questions, for instance, as to whether the composition of microbial communities might change as a response to different pH levels. Ocean acidification is a major factor. So we've constructed mesocosm experiments that help us more or less get at least a more defined response to some of these questions, because there we can, in a controlled environment, vary these different variables, here being pH, and see what impact that has on the structure and composition of microbial communities.
We have a next question, Larissa. Do you believe that increasing the collect cycle sampling or doing multiple times can help elucidate microbiome diversity analysis?
Yeah, absolutely. I mean, one of the things that a lot of the community are trying to do is try and pioneer the use of automated samplers to do exactly this, because it's, as you can imagine, it's quite expensive to undertake some of these sampling expeditions. It costs a lot in terms of man hours and manpower. So if you can construct drones or automated machines to sample various parts of the ocean, it would be very important because the dynamics are, we believe, very different in winter compared to spring, but it's often not possible to have that temporal understanding because you cannot sample at those points. So if you could do it through automated samplers and be able to look at diversity and also functional patterns throughout different time points, I think that will give us a lot of important information. And once we have this information, of course, then we'll be able to make much more direct inferences, which can then feed into models or predictive models, looking at, for example, the effect of temperature shifts on marine microbial communities and their abilities to carry out ecosystem services.
I have a follow-up question on my own, Thulani, if I may. We don't see much changes in terms of richness and diversity among the samples from the spring and winter, or winter and spring. But it's fair to say that, and you talk about the resilience communities, it's fair to say that you're planning to do metatranscriptomics to analyze what is changing in terms of genetic expression. Might be the case that you have the same taxonomic groups and they are always there, but the metabolic profile will be quite different from one sample to the other.
Yeah, I think that's a very good point, because of course, we are looking at the data that I presented, functional capacity as measured, through the collection of DNA samples. There are some logistical constraints on our side because, of course, we are using water from CTD arrays to make some of these measurements. And if you've ever been on a cruise, you know that water is a precious commodity, and so you don't have 20 liters of water to filter to be able to extract RNA and run some of the transcriptomic analysis that would really give you the type of evidence that you're looking at. But again, if we could do that, I suppose you would start to see some of those differences because you might not see changes at a phylum level, but there might be, as hinted by the co-occurrence analysis, some taxa that change in terms of their abundances and that carry out a lot more of the processes depending on the variable that you're observing.
Next question you have, please. Someone is asking, what kind of biotech gene activities are you looking on your mags and why? You commented at the beginning that your group are interested also in exploring biotech applications of whatever community you are working.
Yeah, so we've got an ongoing project. So there's several different biosynthetic gene clusters which we're interested in. One being polyketides. We've got a PhD student who is very interested in PKS genes and their variability in the Southern Ocean. So there's one project where we kind of believe there's presence of a hydrothermal vent in the Southern Ocean, where we collected during actually this cruise that I mentioned samples around there, and they've used genome-resolved metagenomics to assemble different bacteria, and then from those bacteria specifically started characterizing different biosynthetic gene clusters, and in this case specifically PKS. Due to their importance, of course, in terms of antibiotic resistance and so
on. I think there's a long stretch here to ask that, but are you guys thinking about using microbes as a bioremediation process during these changes that is happening, especially in the South Atlantic. We're thinking about that, and in the future, uh, to be able to bioremediate by adding back or trying to shape the assembly. This is science fiction right now type of questions, but the long-term goal that can be envisioned. Do you have comments on that, or it's too early?
Yeah, yeah. I mean, it's an interesting question, but you mentioned the example of microplastics. So of course it's quite difficult to have a direct cause and effect when you're looking at some of these processes. So for instance, if you have an experiment where you are simulating specific microplastics and looking at changes in microbial communities, those changes could be as an effect of different nutrient condition and not necessarily due to the addition of microplastics, right? But we are starting some of those studies in terms of doing a simulated experiment with different types of microplastics. And we've approached it from the point of using the data set to first understand what microbes seem to be present at either high or low levels of microplastic contamination, and then using either hidden markup models to specifically look at degradation genes that could be linked to the degradation of specific microplastics. And by so doing, then you can sort of more or less say, okay, the presence of microbe A and these marker genes that have a capacity to degrade microplastics, and then link that back to the experimental setup to say you've got high levels in one type of experimental system versus another, that these are microbes that could potentially be useful for finding a bioremediation alternative or strategy.
Are your group already constantly monitoring the microplastic concentrations in Antarctica?
Yeah, so we do sample. It's quite logistically difficult in the Southern Ocean because we've done just quantification. There's very little microplastic contamination, but I think this is, of course, because of the ocean circumpolar transport and navigation. That everything probably ends up in Brazil, so there isn't enough residence time in the Southern Ocean to be able to look at them. So.
Okay.
But we do look at that, yes.
Okay. We have time. We're almost right on time. If you have another question or a couple of questions that Thulani can entertain. Congratulations on the awesome lecture. Another question about isn't contradictory that there are evidence of endemic, even though the ocean conditions are not so extreme. So that's
Yeah.
and my question is water is fluid, right? So things can change quite fast.
So yeah, I don't think it is contradictory, and I'll maybe use the findings that others have generated from the Arctic Ocean. Right, I think a recent survey by the Tower Consortium and others found that there is actually a high degree of endemism in the Arctic, and so I mean, for me, it's plausible to think that you would probably have similar levels of endemism in the Antarctic. You know, even old surveys have shown that when you look at the variability of certain taxa, even cyanobacteria, there are distinct patterns at the poles, in the Arctic versus the Antarctic. And as to the question of not so extreme, well, I think that's it depends. So temperatures that are low throughout the year for me, in addition to oligotrophic conditions with low nutrient availability, that is pretty extreme. And so, of course, you would think that kind of selection pressure would then select for organisms that have capacity to survive under those prevailing conditions.
We have a last question. Okay. Someone is asking, can we gather marine biochemical data from different periods, understand the climate change over microbial metabolites?
Let's see if I understand. Well, it depends. In order to measure different microbial metabolites, you would have to do metabolomic analysis, either LCMS, to be able to then relate those metabolites back to specific biogeochemical cycling. But of course, it is a viable approach to be able to do this. But again, I'm not sure whether I haven't seen myself a lot of studies where they've done metabolomics in the ocean, probably because I'm assuming the high levels of salt concentrations may probably be an obstacle when running LCMS and related technical skills. I'm sure it is possible to do it, but I just haven't seen a lot of those studies.
Can you see in your samples during spring and winter big change in iron composition? Do you see
In
that?
there, in there,
Iron, in the iron amount that you have
iron.
in
Okay, yeah. So we've got one other study because the part of the problem with iron is that you cannot use a conventional CTD to be able to measure different iron levels. So you need a trace element clean setup to be able to measure, iron. So we've got a student who actually did iron fertilization study where we did have a trace element clean setup. And you can see very clearly from some of her results that, as you add more iron, you see different and discrete patterns of microbial community structure within your microbiomes compared to samples that have even less iron. So, of course, this more or less hints at the fact that, iron is a strong limitation in terms of the structure of microbial communities.
okay. I think that you are right on time, Thulani.
Right.
So we thank you so much for being here, and in the name of Anchor Program and everybody that is involved, I thank you very much for your time and your help on this workshop that we are starting today. So
Thanks
again,
for the opportunity. Yeah.
thank you so much.
Yes, thank you
Yes.
very much, Dr. Thulani. It was a fascinating talk. It
Thank
was a
you.
pleasure to start the event with such a remarkable lecture. Thank you very much.
With that, then we end the first lecture, and we are going to have a coffee break since our speakers are from different parts of the planet. You allow like about 20 minutes between talks so you can set up everything. The stream on YouTube will continue, so stick that on that, and uh, the members of the table will leave now and we'll be back in about 20 minutes to the next speaker. Thank you, everybody, for sticking around. Thank you. Hello, everybody.
Hello, can you hear me?
Yes,
Hi.
and
Oi, oi, hello.
Hi, welcome back, everybody. So we're going through the next talk of our event, the Anchor event. We are very happy to have here Dr. Georgios Johannes Papas Jr., who is currently a professor at the University of Brasilia in the Department of Cell Biology. He has a very vast experience in computational genomics and had participated in several international genomics consortia, such as the Eucalyptus and the Common Bean Genome Project. Well, he will be talking to us about the introduction to bioinformatics workflow systems applied to bacterial genomics. So, Dr. Georgios, welcome to our event. We are very happy to have you here, you and Felipe both. So you have the word now. So welcome.
can you
Well,
can you
thank.
introduce to us, Georgios? Can you
Okay, thank you so much. Thank you, Professor Krieger, for the invitation. And I will start sharing my screen. And I hope you can. Let me see here.
introduce
Okay.
to us Felipe Almeida from your group?
Okay. Yeah.
Thank you.
Okay, and we also have Felipe Almeida, Felipe Marques Almeida. He's a bioinformatician at the computational genomics group. He's the workflow expert, and we are going to share the presentation. So I'm going to give you an overview of computational workflows, and later on, Felipe will catch on and show some workflows that we've been developing here, okay, at UNB. So let me share my screen here. And I hope you can. Okay. So can you see the screen?
Yes.
Yeah, okay, that's good. So what I'm talking about today is it's an introduction to bioinformatics workflow systems applied to bacterial genomics. Okay, so we work here at the University of Brasilia and at the Computational Genomics Lab. And as I told you before, the agenda will be a brief introduction to computational workflows in biology. And later on, Felipe will catch on. But in my part of the talk, I'm going to talk about the end user perspective. So for biologists, non-programmers, okay, and I'll be naming important technological resources, okay, and then Felipe will show you some nice pipelines, workflows that we've been developing here at our group of computational genomics regarding bacterial genome assembly and annotation. Okay, so let's start. I will, in online presentations, I always like to show you where we are. So Brasilia, in the middle of Brazil. Okay, we're over here, and Brasilia, of course, is the capital of Brazil, and it has very nice architecture, and you are invited to come here. Okay, but let's start with the genome year of biology, meaning that now biology translates to big data. Okay, so a quarter of century ago, okay, in the about 20, 25 years ago. Let's say that Genome Year of Biology started with the publication of this first genome of a free living organism. This was a real big breakthrough for biology itself, and it changed biology. So nowadays, in the 21st century, we are in the genome year of biology. Okay. And okay, in this century, what we have, it's an amazing evolution of technologies and the capabilities to decipher complete genomes of complex organisms and, of course, bacteria. So we have lots of genomes now from humans, okay, to strange animals, so axolotl, frogs, and many plants. And of course, we can delve into our other genome, that is to say, our microbiome, which is very, very important nowadays. But to get to this point, we've witnessed a tremendous evolution of DNA sequencing technologies. Okay, from Sanger-based sequencing, okay, which was, I mean, absolutely dominant in our genome projects before, let's say, 2010, and in the last decade, several new sequencing technologies appeared that changed the game. So you all know about the Illumina, PacBio, and Oxford Nanopore. But the main thing is that, well, we have a market evolution in sequencing technologies that are very important for one thing. They generate humongous amount of data, okay, of raw data, what we call a data deluge. You see, so biology now is in the era of big data. We need petabytes of storage space just to get the generated raw data from genomics projects. Okay, and of course, nowadays we can consider biology as a data science because we are receiving data and we are generating data. And there's lots of data available in the internet, which is a very important thing because we don't necessarily have to generate DNA sequence in the sense that many sequences are already in biological databases. Okay, but this is the whole point. Biology is a data science now. And of course, adapting to DNA sequencing technologies, we have bioinformatics, okay? Because to get all the genomes and all the analytical stuff coming out of raw data, we needed the development of biological protocols, okay? And these protocols were adapted to the generations of DNA sequencing, and they are adapting. Just to let you know, there's this paper here in genome biology saying that technology dictates algorithms. Just for you to know, one very common task in bioinformatics is mapping short reads to a reference genome. Okay, in this compilation here, the authors identified 107 alignment methods, meaning we have 100 different programs to, let's say, do the same thing. First of all, this means that this is still an unsolved problem. And of course, there's another big problem for the end user point of view. Which one of these 100 is the best and that I should use? Okay, so coming back here to big data science, what we have now in the this data science biology, we witness a major focus shift, meaning from data generation. Sequencing now is, let's say, cheap. Okay, it's easy to sequence, but this conveys a problem. Okay, if it is easy to generate data, we have to shift the focus to data analysis. Okay, which is not straightforward. Okay, so I really like this this paper here. So bioinformatics goes back to future, meaning that what we have is that high throughput technologies generate vast amounts of data, amenable, and necessarily they should be analyzed by computers. Okay, and this analytical toolbox is what, like, the bioinformaticians develop and should excel in this type of toolbox. On the other hand, for the end user, the biologist that is conducting experiments, these new biologists should be proficient in the computational analysis field. It's a new field, it's computation, but the biologist has to know that. Okay, so there are differences in goals, perspective, training about these two profiles here. But we should keep in mind that what we need to do is to bridge the gap between complex computational analysis to the non-computational savvy biologists. Okay, so anyway, this data after the computational analysis, so we have to turn the data into knowledge, and the biologist has to know about statistics and about the algorithms, about the technologies that generated the data itself, the experimental technologies, how the data was generated. And in the end, we have to turn this knowledge into understanding. So biology takes center stage now. So what is important is biology. Okay, so this quote from the Nobel Prize winner Walter Gilbert gives a very good introduction about what I said until now. And he said that this flood of knowledge, let's say flood of data, means that the the biologist has to be computer literate, okay, in order to change their approach to understand life. So it's a new paradigm in biology, okay? And when I say protocols, okay, if we generate data from any of the genomic, let's generalize from any omic experimental approach, we generate data that has to pass through several steps of analytical processing in order to get the final result. So here I show you just an example of a workflow in metagenomics. And as you can see, the workflow is different from what, depending on the type of data, we need different computational approaches, different programs, and they are complex in the sense that it's a meandering protocol. You see, we need lots of programs. And to do that, okay, one example of our workflow, analytical workflow, to do that, Linux is the prime operating system for bioinformatics. The vast majority of bioinformatics software is developed and, let's say, targets the Linux operating system. So bioinformatics is Linux. Bioinformaticians love Linux. And when I say Linux, it's Unix, okay? And for us, it's everything about Linux, okay? And there's a strange thing for, again, I'm talking about the end user now. The strange thing is that why we love and how do we work? We work like this, you see? Literally a black box. So command line applications, you see, and here we type in the commands themselves. Okay, so this is very challenging for, of course, the normal user, but with just this terminal, we can do amazing things. For instance, here is an example. What you can say is a spreadsheet is the result of a program prediction of protein domains. And you can see here in the yellow arrows that some IDs are repeated. Okay, so let me just try to, okay. And you can see that these lines are repeated. Okay, so what we want to do is to remove the redundancy. Okay, remove the second lines. Okay, and in Linux, we can do this really easy. Okay, so in the command line, if we type in this thing, this monster, we essentially can remove all the repeated lines. For if this contains 100 lines, 100,000 lines, 100 million lines, okay, Excel wouldn't be able to do that. And with a single command line, with a specific tool in the Linux world, we can remove, identify the duplicate lists, count the unique IDs over there. You see, but this is very cryptic. But of course, it takes center stage in the bioinformatics development. So Linux and programming languages like Python, R, are very important if you want to be a bioinformatician. But if you want to use one of the pipelines, chances are that you are going to need one of or all of these three essentially computational world knowledge. Okay. And of course, there are many others more. I'm going to talk about workflow systems later. But if you see these requirements here on the left, are exactly the requirements for a new kind of professional, which is called the data scientist. Not in the biology world, for everywhere. So biologists can be data scientists now. Okay, and these is important for the biologists of the 21st century. It's very important to highlight that this is fundamental if you want to be a bioinformatician, all of these technologies here. But it's important to know that nowadays, a person that doesn't have all these requirements will be able to run complex workflows, complex bioinformatics workflows, if learning the correct tools and replicating what is in the instructions. Okay, so it's a kind of we're bridging this gap to turn this more amenable to non-bioinformaticians. How? Okay, but of course, going back here, the non-bioinformatician, for the non-bioinformatician, Linux is a burden. Okay, it's command line based. It's kind of a non-intuitive environment. But just to let you know, Linux has the same graphical user interface as the common Windows system with Windows and all the software. You should give a try. Okay, so but one important thing about Linux is the simple fact. Which is a mundane task, let's say software installation. How? This is not an easy task in Linux. It might not be an easy task in Linux, okay? Because one program requires several other software dependencies to be installed previously. There are some other issues, errors in the installation process that definitely sometimes will require trained personnel to solve this problem. Okay, so nowadays we have this effort which is called Conda and Bioconda, trying to make software installation in the command line real easy. Okay, so Bioconda, as you can see here, Conda is a software installation. It's a general thing for general software, even languages, and Bioconda is geared toward bioinformatics software. Okay, so in the command line. So for instance, if you wanna just install NCBI Blast here at the right, okay, there's a command line, but once you copy and paste that, you can install here NCBI Blast Hammer or any other software. Okay, even Kime and so on and so forth. Okay, so what this command does is, okay, I have to install BLAST, and I have to install this enormous amount of requirements, or what we call dependencies, or other software that BLAST depends on. And this is automatic, okay? And in the end, after installation, here we go, BLAST. So instead of having the graphical user interface that you have in the at the NCBI website, you can do in your computer, even if it is just a laptop. Okay, and what is the advantage of that? Okay, so you have your own genome, so you sequenced your own genome. You can format this genome as a BLAST database and start investigating genes and doing BLAST searches in your genome that, of course, is not still deposited in public databases. So this is a vital process in the genome annotation task of let's say, bacterial genomics. Okay, so okay, having known that Linux is the operating system for bioinformatics, we have a problem. The majority of the end users use Windows. Okay, how to cope with that? There are several options nowadays. One is very important. Even Microsoft knows that Linux is the best operating system, and they have what is called the Windows Subsystem for Linux, WSL2. Okay, and meaning that you are running Windows. Okay, you open a terminal, a PowerShell window, and you can essentially install Linux. Okay, and in that window, you are going to have almost all the command line tools and utilities necessary that are Linux. So you run Linux directly from Windows, okay, without the need to have what we call a dual boot setup, meaning that in your laptop, in the when you restart the system, you can select either Windows or Linux. With WSL, you can always it's not recommended, okay, to put in the Windows and then have a Linux Windows where you do the real work. Okay, we also have what we call virtual machines, which are essentially programs that kind of do what WSL does. But WSL works just for Windows. In the case of virtual machines, one free alternative is called VirtualBox. So everything that I name here is just Google VirtualBox. It's very easy. It's a program that can be run in any operating system. Meaning, okay, I can install VirtualBox in a Mac machine, okay, and inside VirtualBox, I can create a virtual machine. Okay, it's a large, let's say, file that will run Linux. Okay, that will run even Windows. So this new operating system will be run in a window. At the same time, as the host operating system. But this is kind of heavy on computational resources. And nowadays, we have a new technology called containers. Okay, so containers are, let's say, lightweight virtual machines that facilitate working inside a host operating system. Okay, you can execute programs targeting different operating systems like Linux. And I'm gonna go deeper into containers now. Okay. So these containers, they provide one thing for us, which is we call operating system agnostic execution. So what is a container? It's a kind of a virtual world, okay, but lightweight, in which, as I told you before, we can execute a Linux program inside, let's say, a Windows machine. Okay, and what is the advantage of that? If all, let's say, the software, the Linux, the bioinformatic software runs on Linux, and I have Windows, containers will close this gap. Okay, and if I don't have to install a Linux distribution, but I can run a Linux program inside my host. So several container technologies are available nowadays. One very important is called Docker. Very important for bioinformatics, Singularity, Podman, Charlie Cloud, all of these you can, of course, Google when they are important. So, what is a container? A container, we call the containers will have an image, which is a file or a series of files that, let's say, embed a Linux distribution, but it can be even Windows distribution. But in our discussion here, these images embed a Linux distribution, for instance, like Ubuntu, and therein you can, the developer can issue commands to install software for the desired analysis. And inside, so at the end, inside this image, we'll have a series and a streamlined, okay, set of Linux executables, okay, of course, focusing on the desired analysis that you have, and even programming languages. So it is a slim, very slim operating system inside some files, which we call image. Okay, and in the host machine, you can execute what is contained in the programs, the executables that are inside this image. And of course, this is a command line, it's complicated, but this simple command line, it, it, it's very important in one thing. You are essentially running Linux inside Windows, and you can run any program and bioinformatics program. So in Docker, you can build containers. Okay, a Docker container saying, okay, from Ubuntu distribution, install. These are Ubuntu Linux, okay, commands, but meaning, okay, install this program, Apache, is a web server, and do several commands. So you build the image, okay? And in the end, let me show you. These two steps, who does that? The software developer. Okay, so the software developer, the bioinformatician, will provide you with a slim container containing all the necessary software to run the pipeline. Okay, and the end user, okay, will download the image, and then, as you can see here, you can run the web server. So keep in mind, this web server is all composed of Linux executables. And here in Windows, I can uh, execute this and have in my machine a web server. Okay. And of course, I can perform, here is a very general example, but you can perform any bioinformatics analysis, okay? And with that, for the end user, we have just to copy and paste the command and zero installation of all the necessary software. This is a very interesting feature that the end users should adopt now. Okay, Docker containers. And how about workflows and workflow management systems? Okay, so here is another example of a workflow. Okay, so we have input data from Illumina short reads, nanopore long reads. We perform a task, okay, assembly. And for this task, we need a program, okay? After assembly, we have to, let's say, polish the faulty nanopore reads. And you can see that there's a concatenation of analytical steps, and there are many of them, and with branching options, which is if we have 1% of duplicated features, okay, genes after these analysis, that depends on all the previous analysis. So if this, we perform this. Otherwise, we go ahead and take this branch of analysis, okay, until reaching the finished assembly. So this is a workflow. We have several decisions. We have several programs that have defined inputs that produce defined outputs, and we have to concatenate, forming a pipeline, what we call. And just to let you know, here I use workflows or pipelines interchangeably. Okay, for me they are the same. Okay, so the workflows and pipelines will be this logical connection of intermediate steps that are required for a comprehensive analysis of biological problem. Okay, and to design a workflow, we need these software components, okay, which are Linux, essentially Linux components, mainly, okay, and we need to know their version, their execution parameters. So, okay, I used an e-value threshold here of 10 to minus 10. This is a parameter, okay? Essentially, what we have in this workflow is a biological protocol. We have to record all these steps, what we call metadata, versioning, which parameters are used in order to be reproduced by others. Okay, this is the importance of bioinformatics workflows. It's not to get the job done for your specific data set, but to be generalizable. And what important is for the analysis itself, not for the data or the species. Okay, so the workflows, as I said before, will involve a chain of transformations of files from individual tasks. So task here is a program. It needs inputs, generates outputs. Okay, and then I link the outputs of task one as the inputs for the task two, and so on and so forth, and until I get the final results. Okay, so essentially what we have here is what we call a dependency graph. Task two cannot be executed unless task one finishes and produces outputs. This is a dependency. Okay, and this is a very simple example. Okay, so just to let you know, okay, so this is a kind of another analytical workflow. So I have input data, a set of FASTQ files, a reference genome sequence, what I want to do. And you can just say what you want to do. First of all, I want to do a quality control. For that, I used the program FastQC. And very important, when you write a paper, you have to say, okay, I did quality control with FastQC using version 0.11 and so on and so forth. And then other step are the tasks. Another program with some parameters here. This minus I is something else. And this is chained together. Okay, so what we've traditionally have done, okay, in the early days, okay, in the last decade, is, okay, step one, okay, I have a command line. Okay, first of all, I have to install FastQC by hand, and then execute FastQC with the data and some options, parameters. And then after the results, I go to step two, and I can say after step one, quality control, I have to discard some samples, okay, because of poor quality. So I can go to the next step, and then I repeat. In the early days, what we did, so workflows were created by, okay, installing individual components in a Linux machine, and after the command line, we just recorded in text files, okay, such a like a recipe, okay, and then you could copy and paste and put all the lines of Linux comments, okay, and at the end your pipeline would be a file, a simple file, so which is unreadable, okay, by known bioinformaticians. So this was cumbersome and error prone. And another problem is that, okay, some tasks take a lot of time. Okay, so manually executing each step and waiting for the test to end. In some cases, okay, this task demands like 3 days. You cannot stand in front of the computer for the 3 days, and then you have to wait. So it's not the execution is fragmented, and of course the software's installation is totally assigned to the user. This is a very bad scenario, which is totally prohibiting for non-bioinformaticians. This is why you always needed bioinformaticians. This is an important point. Okay, so I am a bioinformatician, and what I'm saying to you is that there are some steps that you can do, some knowledge that you can acquire to avoid the necessity of having a bioinformatician at your side. Okay, so here come these workflow composition frameworks. Okay, so of course we have many raw data. We need workflows in modern biology, okay? And in the past years, several attempts were created to mitigate these problems. The, let's say, bioinformatician dependence, okay? And these attempts they are generically called workflow frameworks, and these frameworks facilitate the composition, the building of several tasks, and these frameworks orchestrate, okay, the workflow execution itself. What I mean by orchestrate, they will execute, and these workflows will know when a task ended to start the dependent task. See, so in the workflow framework, it triggers the execution of step one. The workflow system knows when step one ends, the files generated by step one, and feeds it to step two and starts step two to until it ends and the process repeats. So this is the workflow orchestration, and the end user don't have to care about this. Okay, this is very interesting. So we have several workflow composition frameworks. We have graphical ones, which are very important. Frameworks based on programming language scripts. And in this case here, you need Python or R knowledge. And this is kind of advanced. But there are several new frameworks that create a new programming language, but it's a very easy to read language, which is what we call DSL, or domain specific language. It's a language that provides a clear definition of workflows, individual tasks, the inputs and the outputs. And I will show you some examples later on. Okay, so I invite you to read this paper from 2021 from, of course, bioinformatics. It's a methodology in biology. Okay, so Nature Methods publishes several workflows. Okay, and what we this paper provides is a compendium of workflow management systems. Okay, and the advantages of the workflow management systems. Essentially, what these frameworks do and what they have that's very important to for the work is that they provide reproducibility, portability, which is interoperability, meaning I can use execute the workflow even though it's based on Linux tools. Given containers or conda installations, I can execute things in Windows. So this is what we call portability. Okay, versioning, fault tolerance, which is very important. If a task somehow gives an error, okay, so I have 10 tasks. The workflow executed for 9 steps. And in the very last one, it gave you an error. But this, until the running 9 tasks, it took like 3 weeks. Okay, in the old days, we had to essentially execute from ground level, from zero. And here with the workflow systems, we have checkpoints, meaning that the execution can be restored from that point on. Okay, these systems provide a simple, okay, for execution and configuration, and they are very easy to share. Okay, one important tenet that I told you before, the workflows should be shared. They should be reproduced by others with different data. Okay, and another 2 points here. We have resource management. The workflow systems, they manage your CPUs, the memory that your machine have, and it can scale from a laptop to a very big server with hundreds of processors. Okay, so the workflow systems will do that. Okay, so here now, what we have with workflow managers is that, okay, having the pipeline installed, the necessary programs, they are installed, let's say, in containers, in conda environments. So it's very portable. You can execute from your computer to a cloud computing platform, and you can see the names here: containers. Okay, resource management. How many CPUs I'm gonna use, and we have checkpoints. Okay, if this here provides an error, and these step 1 and 2 were successful, we're going to execute only step 3. So it's a very big improvement. So in the paper, there are several names we have several frameworks. You have to use any one that you are best suited. Okay, so graphical and systems with DSLs, okay, domain specific languages, and other ones. And here I'm gonna just highlight these 2 based on DSLs, and these workflow systems are called Nextflow and SnakeMake. Very important in bioinformatics. So record this SnakeMake. It's a workflow management system. Of course, it's reproducible, scalable, and nowadays it's highly popular in biology. So there are several, let's say, more than 7 citations per week using SnakeMake to enact and construct a workflow. Okay, here's the publication here, and workflow provides a syntax for a task. Okay, this is what we call the DSL. Okay, domain specific language. So rule is the task name. Okay, and a rule is the task, and the name here, you provide a name, BWA map, and in this language it provides clearly the inputs, the input files, what it generates as output, and the, when you see shell, is the Linux command used. I'm not here to put this in perspective for you to learn the DSL, but after a while you can learn this even though you don't have programming experience. Okay, so of course, and then you can expand the rules and create a pipeline. You see, so BWA map is a task, and then it generates the output that is used by some to sort, and then SnakeMake can, given this description, which is created by the developer. Okay, so the developer will distribute this pipeline, and the end user will run it with the input data. Okay, but of course, what is important here, I'm not saying for you at this point to be a developer, but be a user of SnakeMake pipelines. So there are several SnakeMake workflows here, and you can see there's a SnakeMake workflow catalog. Let's just Google it, and you see there are, let's say, 60 different workflows, ready-made workflows. If you want to do a transcriptomics analysis, okay, just download this workflow, and you are gonna need your input data. And there are several workflows for metagenomics. You see, just get the name here. If you click in the link, the developer will provide the, let's say, short manual of how to run the workflow with your data. Okay, so there are several metagenomics SnakeMake ready-made workflows, and you can use that. We use this. We published a big workflow for single-cell RNA-seq analysis made by Wendell. Wendell was a former student at UNB, and now he's at the University of Florida. And what we use here, okay, the names of the technologies: SnakeMake, Docker, and some programming languages to provide a workbench of analytical steps. And the important thing, to install this and run the pipeline, the end user does absolutely nothing. Okay, absolutely no, it's almost nothing. Okay, and just to end, we have Nextflow, which is another very important player in the workflow world. Okay, remember this name. Here is the website, Nextflow. Nextflow has a different domain specific language, the DSL here, saying that here we have a task, say hello, it has inputs, generate outputs, execute this command, and the workflow. The language itself is not important in this context, but Nextflow provides exactly what we commented before: reproducibility, portability, checkpoints, and so forth. Okay, so there's this website here, Nextflow Core, NF Core, which is a community effort to host curated analytical pipelines using Nextflow, meaning that NF Core, there's a publication for that. There are several ready-made pipelines. You see, if you want to do an ancient DNA analysis pipeline, so developers already have put all the components inside this, the containers, the Docker containers, Singularity or Conda, anything, and you can just concentrate in providing your data. Another one important for metagenomics people is this AmpliSeq pipeline, which is very interesting. It's easy to use, but you have to read the manual, okay? And it contains several steps, analysis. I'm not going through this. And there are several other pipelines dedicated to bacterial genomics. So this MAC pipeline, it's assembly and binning of metagenomes. There's the BACT-MAP for assembly of whole genomes. But I will end here to let Felipe, I think I took a lot of time here, but I'm going to let Felipe show you some bacterial genomics pipelines using Nextflow, okay, that are very easy, and the end user will be able to do any bacterial assembly and annotation giving the data, and in a laptop. So I'm gonna end here and see if Felipe is available.
Yes, here, try to share screen.
I'm gonna stop sharing the screen.
So can you see it?
Just a second. Yes.
So, okay. So I'll continue here. After this marvelous talk from Georgios, now you actually know what pipelines are and the differences of pipelines or workflow management systems from just command lines put in a file. And I'll try here to just give you a quick example of how easy it is for users to use these build pipelines. Like, it's difficult to build because you need to be a developer, but for the user, it's very easy to use. So these are the three pipelines that I'll show to you. They are all open source and hosted in GitHub. They have been built and are still maintained by our laboratory by us, Georgios and I. Until today, we are still enhancing these pipelines whenever the users use it and have ideas for it. And the pipeline has been implemented using Nextflow, mainly because it enables the seamless connection with Docker images, meaning that users need no installation at all. They just need to install Nextflow and Docker inside their machines. And whenever an image, another Docker image is needed, Nextflow automatically downloads it and uses it without the user having to do anything. So it's very reproducible, as Georgios already said, and it's standardized analysis for 1 to 1,000 genomes, meaning it's the same command whether you have one genome or have 1,000 genomes, have 10,000 genomes, etc. It's literally the same command line. And it has an easy parallelization, meaning it also enables you to run in from laptops to high cluster machines. And it's very portable, as here had said, because it uses Docker, it uses Singularity, for example. You can execute Nextflow in any machine that actually accepts these containers like Windows, Mac, and Linux. And so it is very easy to install, and it provides a cross-platform execution. And Nextflow, these pipelines, they actually enables us to create complex workflow for genomics, for instance. And this is the workflow that can be built while using when using these three pipelines together. We start from raw genomes, from raw sequencing data, and preprocess this sequencing, perform some quality checks, trimming adapters, demultiplex it if it is multiplexed, all right, and use these preprocessed reads to generate assemblies. So what is beautiful from these workflow systems is, for instance, in MPEG-app, which is our pipeline developed for genome assembly, we have 9 different assemblers, meaning that if users would actually want to have 9 assemblies with different software, they would have to execute it 9 times. But with a workflow, the 9 different assemblies are already inside the pipeline. So with a single command line, you already have the 9 different assemblies and the comparison between them. And then you can pass it the best of which you choose to the annotation pipeline, which is called Backanot. So they are all platform independent. They accept Illumina PacBio Nanopore reads. And what we actually have to highlight here is that they are fully documented. So users can just go to the manual, to GitHub itself, where the manual, the documentation is there, and they are very extensive, meaning if you never heard of it, you can read the documentation, execute the example data sets we have there, and you directly understand what to do. So I'll just show an example. For example, to preprocess the data, you can see now it's not properly formatted, but with a single command line, we call nextflow run, the name of the pipeline, which is the GitHub tag. That, for instance, for this one would be fmalmeida and Jess preprocess. We choose the profile we want to use for the installation of tools. In case we have available profiles for Docker and Singularity, which one you would like to use. And you just pass the short reads or the nanopore reads for it. And the pipeline will do everything for you. It will download the images, we execute the tools inside the images, and then just give you the results. And what is good about Nextflow, it tracks the execution and it shows to you how many times each step took to run, how much memory it took to execute, so you can actually manage how much analysis you can run parallel or not. And you can see here that it was actually fast. This was run in my laptop. It just took 12 minutes and no more than 3 gigabytes of memory. And here are the expected results. You can see that we receive a directory having all the preprocessed reads, and the quality information of the reads are also given together with the preprocessed reads. And these preprocessed reads are ready to be used in the assembly pipeline, which the same way, it's just a single call. You can see that the command line is always simple because you do not need to configure the tools itself. You just need to pass the data for the pipelines. So here, the main input is a sample sheet which actually tells you again, I don't know why it's not properly formatted, but you can actually see here that the sample sheet just tells you what will be the name of this analysis of this sample we are running, where to find the Illumina reads, for example, where to find the nanopore reads, which strategy we want to execute, the genome size expected for this genome. And as you can see here, I'm just telling about one sample, but in this sample sheet, we can put 10, 20, 30 thousands, and the pipeline will read all of them and execute them automatically for you. And the same way, we have a report here. Here is just want to show you that the most memory consuming tool was Unicycler, and it still got only 12 gigabases, and it can be executed in a laptop. Because laptops today, most of them have at least 16 gigabytes of memory. So you can actually see that is easy to execute in laptops. And these are the expected results. As I said, the pipeline we execute for each strategy, the proper software for each strategy choose. Here, we performed a hybrid assembly. So from the 9 different assemblies found inside the pipeline, just these 3 are built for hybrid assemblies. So the pipeline executes these 3 and gives you a comparison between the assemblies so you can choose the best for you. And it gives you a text file like this comparing the results, and also a file that can be opened in the browser built with MuteKey, where you can see graphs about the same information found in this text file, but just in graphs. And now the same thing, just to exemplify how to execute the annotation, for example. It's the same way as the assembly one, just the minimal required inputs here are the sample sheet that describes the reads, the genome assemblies, or the reads to assemble and annotate, because this pipeline can also automatically assemble genomes for you, and the path to the databases that are required for you. So you just need to execute, for example, next for run backend get DBs, and the pipeline will first download the databases for you. And the same thing, just to show you that it didn't even pass from 4 gigabases of memory used. And these are the results. You can see that a vast amount of different software has been used to provide you a comprehensive overview of your genome. And you can see this is the general annotation built done with Proca, but you can also have like secondary metabolites annotated with AntSmash, insertion sequences, the annotation of genomic islands, the integrative and conjugative elements. You annotate plasmids, multilocus, MLSTs, prophages, and etc. Resistance and virulence, for instance. So here I'm just exemplifying that what is best from this pipeline is that it gives you users annotation files that can be opened in browsers like this one, and also a genome browser and a Shiny app. So it is easier to users to run across the results and to compare the results, to filter, to query it. So here I'm just exemplifying the report of resistance genes annotation. Is inside the pipeline, resistance genes are annotated with ResFinder, RGI, ArgMiner, AmpFinder, and also by Proca itself. And here is just an example of the virulence reports. We can see that we can give graphs about all the virulence factors that are found inside the pipeline, how many genes of these factors are found inside it, and a table with detailed information. And each gene is directly linked to the database, so you can actually understand more about this gene that has been found inside the database. And the pipeline also gives you all the annotation, literally all the annotation built inside the genome browser. So you can actually navigate through the genome browser to visualize all the annotations that are found in your bacterial genome. and where they are positioned. And the best from it's finally the annotation also this pipeline also gives user a Shiny app that is all packed inside the Docker, so you don't need to worry about installing R and things. And this app gives you an interactive report, a way to go across all the annotations found inside the pipeline, that are resulting from the pipeline. You can query your genome again, visualize the genome browser, perform a custom blast. Like if there is a gene that hasn't been annotated by the pipeline automatically, you can use this Shiny app to perform a blast against it and to visualize whether it is found in your genome or not, to perform to filter information you like, and etc. And here are just two studies that we have published in the last year. So you can actually get a gist about what are the capabilities of the pipelines, and what if we can extract the type of information we can extract from the results, and why it can be useful for. These two studies have the results of it have all been built using these three pipelines. That's all. It was just a quick chat for you to know how simple it is to execute these pipelines, because you don't need to install anything. You just need to show where your data is in your machine, and it does everything for you and gives you a beautiful like interactive report, a way to understand the results.
Perfect, Felipe. I'd like to thank you a lot for this wonderful presentation. And so with that, it's a clear example of pipelines in use. And the thing is that Felipe can tell you, I mean, essentially, we can assemble and annotate a genome in a laptop, provided that you have sequencing data, and in any type of sequencing data. So this is marvelous. And what we've done, and Felipe is, let's say, a workflow magician. He's the expert here, I can say, in Brazil. What it can be done is that, I mean, let's say, take from the end user this bioinformatics complexity, and the end user will concentrate on biological analysis of the data. Okay, and with that, we'd like to thank everybody, and we are open for questions, if any.
Thank you so much, Georgios and Felipe, for the great, fantastic lecture. I'm sure everybody appreciates your time and explanations. And I'm sure we're going to have some questions. We're almost out of time for the next speaker, but we have a couple of minutes to convey a few questions. And I will ask Annalisa to show them from the audience, if possible. Yes. So the first one is appearing on our screen. Since the study of omics to understand the web dynamics of microbial marine community, can you use this data to infer the dynamics? Oh, this is the. Oh, sorry. This is the previous talk.
Mm-hmm.
I was reading from the previous talk. That is the new one. Well, I will ask the first two questions. Okay, it can be for both of you. So
okay,
it's displaying now.
Oh, it is.
So, very good, Jessica. So, do you need to know, as an end user, any programming language? From what we have, zero zero knowledge. Okay, but if you are interested in investing in analytical tools and expanding the analytical analysis, uh, uh, in the future, you might be interested in learning some programming, okay? But from now, this is the important take-home message. With these workflow systems, SnakeMake and Nextflow, you essentially don't need to know any programming language, any let's say Linux knowledge also, so you can just execute. Okay, so this is important, but there's a downside on that, because the workflow will be a kind of a black box for the end user. Okay, so not necessarily the analytical steps that are packed inside the workflow will suit for your data. And eventually, you are going to have to modify and adapt new tasks for that. But this is the other step. Let's say you want to enlarge Philippe's pipeline. It is possible, but I mean, this is a long shot. Okay, for the end user, you don't need to know anything.
So, Georgios, I have a follow-up question then, if I may, since I'm chairing this section, okay? The question is fairly simple. You talk about different workflows and have a biological question. You have sometimes dozens or even more workflows to try to answer that specific question, okay? And how a student or someone that is beginning to use these tools will be confident that the output at the end is the right one. I know this is not a right or wrong one, but you can see that if you have some different pipelines of workflows that give completely different answers, you already raise a red flag, okay? But most of times, or many of times, you are going to have outputs that are slightly different, okay? So one workflow will present like give you, as Felipe just show, a gene that's related to antibiotic resistance. And then the next workflow will not show that. So then you will want to write about it and describe that, which would be the right one, okay? Is this gene present or absent? Because they're using different, the different pipelines to give you different answers. I know you explained very well that they're going to, of course, in the materials and methods a lot of you have done, and that in very detailed process, and this is the output. Okay, so it's kind of you must reproduce that anyone and arrive to the same result. That's fine. But when you are going to run two different workflows and give it slightly different answers to that, how it works.
This is a fantastic question, and everybody has to listen to what Professor Krieger said, because bear in mind, computational results are not strictly the biological truth. Remember when I told you, okay, for read mapping, there are 107 programs, different programs. And the question is, which one is correct? Because essentially, if I run 3, I can get 3 different results. Okay, so first of all, in terms of the analysis, you should bear in mind that what looks like rocket science, bioinformatics and programs, it's they are approximations of biological realities, okay, which should be analyzed in the context of your problem. You see, so essentially, antibiotic prediction. We're going to have different results depending on the databases used to scan the results. So the end user should know which databases are the most important, which databases are updated, you see, and which tools, which programs are used for this. So I'm trying to say use the workflows, but don't take the answers that the workflow gives as a ground truth. They are not the truth. You are the biologist, and also, as Felipe said, oh, okay, it's easy to run the workflow. All the end users should know which boxes are being used in the workflow. So it's not a matter of using the workflow, but you should know the boxes and which data it generates. Okay. So, and the last thing, how to cope with this problem. Not necessarily, let me rephrase that. Necessarily, the programs don't have the same answer. What we do and what Felipe did when he said, oh, okay, uh, we use not one program for genome assembly. We use 9 different, okay, and give a report comparing the assemblies. So we don't trust any software because it will depend on your data and so on and so forth. And for all our pipelines and other pipelines, what we do is for the same analysis, we try to repeat it with 3, 4, 5 different programs. And then what we have is a report in another pipeline that we were discussing yesterday. We report all 5 analyses and say, okay, the intersection of the analysis in terms of, okay, they are matching is that. Then it's up to the user to learn which one to go on with the in terms of extracting biological data. So this is the point. What we do is necessarily we do several analyses and we don't trust them. And the end user shouldn't trust bioinformaticians.
Okay, that's a good one. So, guys, thank you very much again. And in the name of Anchor Program, I really thank you for your time. I know in Brazil, here is a holiday, so I really appreciate your time and effort, and really enjoy the lecture. And if you have more questions, we'll convey later on for you guys to answer on YouTube if you were kind enough. Okay, so we are out of time. Our next speaker is already here. So
Perfect.
thank you again.
Thank you so much.
Thank you.
Thank you. Thank you, everybody.
Thank you very much.
Bye-bye.
Okay, so after this amazing talk, we're gonna have Dr. Matt Ohm here with us.
Hello.
Welcome, Dr. Matt. Hi. So I'm gonna do a brief introduction of our next speaker.
Yes, sir.
Dr.
Yes, sir. Hi. Wait a second. Hey, Matt. It's been so long. Before Dr. Juliana introduce you, thank you very much for your time, and I really appreciate you being here and help us with this workshop. So
I'm very glad to be here. Thank you for the invite.
yeah, great. So Juliana, go ahead. You can have the word.
Great. Yes, welcome, Dr. Matt. Amazing to have you here, like Dr. Krieger just said. Dr. Matt Ohm is currently a postdoctoral researcher at the Justin Sonnenberg's lab at the Stanford University. His PhD was from, he did his PhD in the laboratory of Jill Bansfield at the University of California, Berkeley. And he's currently studying the human microbiome and the microbial population genetics, and have developed several open source programs and bioinformatics tools for analysis of genome-resolved metagenomic data, which is amazing for us. I'm sure everybody will make the best out of this amazing time that Dr. Ohm is giving to us. So today he will talk about the genome-resolved population genomes and string level comparisons. Dr. Ohm, welcome again.
Thank you for having me. And thank you for the introduction. Let me go ahead and share my screen now. Let's see.
Sorry, everybody. We apparently we had a problem here with the connection. So let's just wait a few moments for Dr. Matt Ohm to come back. Yes.
Sorry, I'm back. My Chrome restarted, but it looks like I have everything working now.
Great, great.
Apologies for that. Okay, is my screen working okay? Can people see what I'm sharing here? Yes, wonderful. Okay, I'll get started. Thank you all again for that introduction. My name is Matt Ohm. I'm a postdoctoral fellow at Stanford, and I'm very happy today to talk to you about some of the work I've done doing genome-resolved population genomics and strain level comparisons. So by way of introduction, I want to start and give very, very broad historical context for what I'm going to be talking about today, which is, again, genome-resolved metagenomics. So what you're looking at right here is the first ever published photograph of a pure bacterial culture. This was taken by Robert Koch in 1877 on the right here. This is Bacillus anthracis. And prior to this photograph, people knew that bacteria caused disease. Robert Koch was a contemporary with Louis Pasteur, but people thought bacteria in general caused disease in general. And what Robert Koch did that was so impressive was isolate out a single bacterial strain from the milieu that bacteria exist in, and isolated it and characterized it, these pure bacterial strains for the first time in culture, which was very revolutionary. And so that was done in 1877. And in 2004, Jill Banfield did pretty much the exact opposite of that. Whereas Robert Koch famously was able to interrogate single bacteria, in isolation, one at a time. Jill Banfield in 2004, for the first time, sequenced all of the DNA of the entire community all together at once. And this, what made this so revolutionary is what Robert Koch did not know is that only a minority of bacteria are actually able to be isolated and studied in pure culture like this. And 90% of bacteria are resistant to culturing. And so when Jill Banfield did metagenomics for the first time, specifically on acid mine drainage, which was a very simple community, for the first time it allowed scientists to study bacteria without the need for isolation. And so Robert Koch's discovery led to what was called, or what is referred to as, the golden age of bacteriology, which is a 30-year period in which all the principal bacterial agents of human disease were discovered. It makes sense that this was entirely revolutionary in science. This is still the foundation of modern biology, right? Petri dishes, isolation, studying bacteria. And similarly, Jill Banfield's discovery in 2004 led to a complete restructuring of what we know of as life in general. If you Google the tree of life on Wikipedia, you'll find this phylogenetic tree, which was created by Jill Banfield based on the genomes that we know of from metagenomics. Each of these clades that has a red dot on it is an entire clade of life that we only know of thanks to genome-resolved metagenomics, because these clades cannot be isolated, at least as of when this tree was made in 2016. And if you look at most recent snapshot of GTDB, which is a database of all bacterial species known, for the first time very recently, more than half of all of the species in GTDB are exclusively known through metagenomics and not culturing. So, in the 20 years or so since Jill Banfield from the forced metagenome, we discovered more bacterial life than in the 150 years since Robert Koch started isolation. And so with that kind of historical context with metagenomics, I want to talk a little bit more about what exactly I'm talking about and comparing it and contrasting it with other methods. So if you have a microbial sample that you want to study, there's kind of like three main options here. The first, which is what Robert Koch would have done, you can isolate individual members from that community into pure cultures and sequence these pure cultures in isolation if you're in the genomics era, which we are. And so you get these isolate genomes of individual pure cultures. An alternative way of studying that is with amplicon sequencing or 16S sequencing. This was pioneered by Carl Woese in the 70s. And in this scenario, what you're doing is you're amplifying out and sequencing just a single gene from the entire community. Usually it's the 16S rRNA gene. And in doing this, you're skipping this isolation and culturing step, which means you can study everything in your sample with the 16S gene without requiring isolation, and do these very detailed and interesting phylogenetic trees or amplicon-based analyses on these amplicons. This was a huge revolution, and it led to the discovery of archaea as another domain of life. But I hope you can realize that it's a little bit limited in that you're only sequencing one gene from the community and all members of the community, but you don't know anything else about what the rest of the genome is that of this gene that you've sequenced. So you can find a very cool variant of a gene, but you have no idea what the life is from. And what I'm going to be talking about, shotgun metagenomics or genome-resolved metagenomics. Rather than amplify out a single gene, you sequence all of the DNA in the sample in what's called a metagenome, and then use computational tools to assemble these short reads into metagenome assembled genomes, or MAGs. And this technique, again, was pioneered by Jill Banfield, which is where I got my PhD from at UC Berkeley. And so today, metagenomics is used for a lot of things. The three primary use cases that I see in the literature, again, where you start with your metagenomic reads, are taxonomic classification, functional annotation, and genome recovery. I've listed just some of the many tools that are available to do each of these tasks. Taxonomic classification is if you do metagenomics on a community and you want to know what are the species present there, what types of taxonomy is there. And so there's a lot of tools that are able to do this. Most popular, arguably, in the human microbiome field are Kraken and Metaphon, etc. Functional annotation is if you have these metagenomic reads and you want to know what sorts of functions do they have, like carbon fixing, acetate metabolism, whatever it is. And there's a lot of tools to do that. And genome recovery is becoming more and more a task that people want to perform, which is where you assemble these short reads into longer contigs, and then you bin these contigs into genomes, essentially to discover new life. And so, yes, there's another of other tools that can do that. What I'm going to be focusing on today, however, is a kind of subset of metagenomics that is it's more experimental and kind of on the cutting edge. These types of things are not yet easy to run necessarily with the types of like pipelines that are going on, and they're more rare to run. My current postdoc advisor refers to this as artisanal metagenomics, but this is if you want to spend more time to kind of learn how to use the computer and how to run these bioinformatic analyses, these are yes, they're kind of more fun and artisanal and out there. And so what I'm going to be focusing on today for this talk are these specific aspects of artisanal metagenomics: strain tracking, looking at types of life that is not bacteria, looking at in situ replication rates, again, looking at function here, as well as focusing on microdiversity, which is not very well studied, but I'm absolutely fascinated by it. I spend most of the time on strain tracking and microdiversity, but I will talk about all of these aspects. Okay, so getting right started with intra-individual diversity, microdiversity, in situ population heterogeneity. All these concepts all these names refer to the same concept, which is that when you're studying a natural bacterial population, you're never actually having a clonal population of cells. So for example, even if you take a single cell of E. coli and put it in a culture flask and grow it up, by the time that single cell multiplies into the billions of cells that are necessary or that constitute a dense bacterial culture, each time a bacterial replicates, right, thanks to non-perfect DNA polymerase, you end up with little mutations in the daughter cells. A random chance of a mutation in any of these locations. And after enough generations of bacteria multiplying, you end up with some of these mutations being deleterious, some of them being selected for, and you end up with what sometimes is called a strain cloud of diversity, which is a bunch of bacterial cells that are all highly related to the founding population, but are all slightly different. Each two individual cells in a dense culture of E. coli likely are not identical and have small differences between them. And this isn't just true in culture flasks. If you look at natural bacterial populations, you find somewhat substantial levels of this microdiversity. Again, as was mentioned in the introduction, I primarily studied the human microbiome, so that is where most of my examples are going to come from. And these are two kind of beautiful examples of this concept. In this study here at the top, this is a study done on Bacteroides fragilis, which is a common member of the human gut microbiome. Each color on the outside represents a single individual, a person, and each leaf is an isolate of Bacteroides fragilis from that person. And what you can see is that each person has their own distinct population of Bacteroides fragilis, their own strain of Bacteroides fragilis, but also within an individual person, all of the isolates are not the same. Again, showing that there's this microdiversity present in the population. And you see the same thing with HIV particles within patients' blood. In this case, each color is a different individual infected with HIV, and each leaf or node at the end of the tree is there's the sequence of a different isolate of HIV from that person's blood. And so again, you can tell each person has their own population of HIV, but the two HIV molecules within the blood are not identical to one another. And so kind of the last thing I did while I was a postdoc in Jill Banfield's lab was write this program in Strain that is specifically designed from the ground up to measure and analyze this microdiversity present in bacterial populations. And so some of the examples of things that you can do with this, it's essentially the way that you measure it is by looking for mutations present in the reads that you get when you do metagenomic sequencing and align them to a genome. And so just by kind of if you scroll over a mapping file, you can see, okay, well, there are regions of the genome that have low nucleotide diversity, so there aren't many variants present. And there are regions of the genome that have high nucleotide diversity, so there are many variants present. You can also measure things like the density of variants, single nucleotide variants, SNVs, how coverage varies, again, measuring nucleotide diversity, which is just a measure of essentially how snippy the alignment is. You can also look at things like linkage decay, which has to do with recombination frequencies. You can look at site frequency spectra. There's a lot of things you can look at with microdiversity, some of which are programmed into use with InStrain. And I will, at the end of the talk a little bit more about the practicalities of running this program and doing this type of analysis. But for now, I kind of want to spend a little more time emphasizing what it can do and why I think this is so cool. And so first, microdiversity analysis directly led to the discovery of CRISPR. For those of you who don't know, Jill Banfield discovered CRISPR and then told Jennifer Doudna, hey, you should check this out in human cells, which of course led to Jennifer Doudna getting the Nobel Prize for characterizing CRISPR very recently. And so the way that Jill Banfield identified the CRISPR array was she noticed that on bacterial genomes, there was often a region of the genome that was very variable between the different cells from one another. That is, it had microdiversity. In these early metagenomes, it was noticed that the CRISPR array was very variable from cell to cell. And so following up on that led Jill Banfield to discover CRISPR and then tell Jennifer Doudna about it. So anyways, a nice little anecdote of why microdiversity analysis is cool. Another example has to do with COVID-19 and wastewater. So I was lucky enough to be involved in this project early in the COVID-19 pandemic, where we did metagenomic sequencing of wastewater looking for COVID-19. And so I think everyone probably knows by now, but people who are infected by COVID-19 actually shed viral particles in their stool. And so if you go to like a municipal wastewater center, you can look for COVID-19 genomes and get an idea of how much COVID is circulating in the neighborhood that's feeding into the wastewater where you're looking at it. Typically, this is done with qPCR, so you're just quantifying how much viral load is present, to know how the outbreak status is. But in this case, what we did was we actually did metagenomic sequencing on the COVID-19 DNA to see if we could tell what variants were circulating in the population, again, in the wastewater that we were sampling. And so without getting too much into the details, this project was extremely successful in our ability to identify circulating variants that were present. So essentially, by doing a metagenome of these different populations around the Bay Area, specifically Oakland and Marin, we were able to, if you do metagenomics of wastewater and look for the variants that you see in the wastewater, these turned out to pretty much perfectly match the variants that are actually circulating in the community as detected by traditional COVID-19 sequencing tests, kind of highlighting the fact that by doing wastewater sequencing, you can identify the circulating variants in the population. And I want to point out that this study was performed and published very early in the COVID-19 pandemic, as we know now, September 2020, which is before anyone really cared about variants. This is before Delta, this is before Omicron. When we performed this study, it was purely an academic question of whether or not we could identify COVID-19 variants. I mean, we just used InStrain. It's not like we were using, specifically developed tools. Later on, this turned out to be extremely important because, as we know, variants of COVID are something we all care about. But this is just an early proof of concept showing how cool microdiversity is and how cool the analysis is. And you never know how important it might be later on. And then kind of the last example I have has to do with the ocean in particular. This is one of my favorite studies of microdiversity analysis that was performed by Murat Aaron's lab in 2019, where they were looking at variants present in SAR11 genes across the ocean. And they identified many different genes that had microdiversity present in nucleotides of those genes that affected the folds of these genes that they were able to map, across the ocean. And so I don't want to get into the details here, but I just want to point out that this type of analysis, InStrain isn't the only program that can do this. Anvio has a proven track record of also doing this type of analysis explicitly in the ocean. And it's all kind of the same thing and all very cool as far as I'm concerned, this microdiversity stuff. Okay, wonderful. So that was talking about microdiversity. I'm now going to shift gears kind of a lot to talk about strain level comparisons. This is extremely relevant and common to do in the human microbiome, where you're looking for transmission of strains between people or between people in hospital rooms and outbreak settings, et cetera. But it's applicable to all environments where you're doing metagenomic sequencing. And so, yes, so to talk a little bit about, introduce this question. When you're doing strain-resolved analyses, or you're trying to track strains between different metagenomes, it's very important that you have a threshold to distinguish if these two organisms are the same or different. And there are a number of different thresholds that have been established based on average nucleotide identity. But I find it difficult to talk about these in the context of invisible microbes, and I like to take it to macro world and talk about dogs. So for example, if for the purposes of strain tracking, you say that two organisms are the same if they're of the same genus, that's like saying a beagle and a wolf are the same because they're the same genus. You know something about sameness. Like, it's not like you're comparing a frog and a fish. These are the same. They're very similar organisms, but be very difficult to say, I have the same organism, a wolf and a beagle. They're very different. You probably want something more stringent. And in the microbe world, that would be like saying doing strain tracking based on 16S sequencing. Saying, okay, I say the same 16S sequence in these two places, which means it's the same genus in these two places, so therefore it's a strain transmission. That's probably too broad for the context of actually tracking strains. You can go a little bit more detailed to the species level. In this case, we're now all talking about dogs, but again, these dogs are pretty different from one another, and we probably want something more similar, more stringent thresholds. Getting to the strain or the breed level, strain is the concept in microbes, breed is the concept in dogs. Now we're getting somewhere, right? These dogs are all very similar. If you're saying these two dogs are the same, I understand where you're coming from. They look the same, but at the same time, these are not actually the same dog. These are actually all pictures of dogs I had growing up. They're all German Shepherds, but they're different dogs. They're highly related, but for the purposes of strain tracking, if you're trying to trace an outbreak or something like that, these aren't the same. What we really want here is a level of stringency that can get us to identical. And so in this case, this is Barbara Streisand's dogs, which she had cloned. And so in this case, you're actually looking at two of the same dog because the dog was cloned. And so that's the level of stringency that we want when we're doing these types of strain tracking analysis. And so the question is, okay, well, what is the stringency required to differentiate between organisms that are identical? And so again, looking at these thresholds that have been established for microbes, as I said, 16S sequencing is about the genus level. The species level is about 95% average nucleotide identity. I should talk about ANI for a second. ANI is a very simple metric to measure how similar two microbial genomes are. All you do is you just align the two genomes and then you count how many differences there are between them. It's that simple. So an organism that has 95% ANI, which is average nucleotide identity, means that for every 100 base pairs that's aligned of the genomes, there are 5 differences. So it's 95% the same. And there's a lot of work that has gone to show that the bacterial species differentiation is about 95% average nucleotide identity. In terms of strains, there isn't a consistent threshold. Different people use different thresholds. Different taxa have different thresholds that are commonly used. It's not a stringent term. But in terms of identical, there isn't really a threshold established. You could imagine it should be 100% the same. But as we just talked about with microdiversity, it's very possible that as soon as a daughter cell splits, it could have a mutation. So you probably need some sort of wiggle room in there. And so we kind of decided, okay, well, let's call two organisms the same if they've been diverging from one another for less than 10 years. So, they're highly, highly, highly similar, but give them a little bit of wiggle room in terms of, DNA polymerase not being perfect. So then it begs the question, okay, well, how much can a cell mutate in 10 years? So for that, we checked out a couple of different, studies looking at how fast bacterial populations tend to evolve in the wild. And it turns out, so these other thresholds, same 16S cluster means you've been diverging for more than 250,000 years. Even at 99% ANI, in order for two organisms to differentiate that much, it means they've been diverging for about 44,000 years. And so this 10 years divergence, turns out you need a ANI threshold of something like 99.9999% average nucleotide identity. And so for doing this like detailed high resolution strain tracking, this is the threshold we wanted to catch and be able to measure when differentiating these identical strains. But this turns out to be a difficult thing to do, specifically because of this microdiversity that we talked about. When you're doing strain level comparisons with metagenomics, you're not comparing one genome to another genome. You're comparing, one read cloud to another read cloud. As we said, in natural populations, there's all of this strain diversity going on in these strain clouds. And so the question kind of comes down to, if you want to be very precise and accurate with your genome comparisons, when you have microdiversity, it kind of complicates that. And so, how do you do these microdiversity comparisons at an accurate level? And so, some approaches that have been taken are, just isolate a bunch of sequences out and compare those. You could maybe just compare the consensus genotypes. So at each position on the genome, let's just compare the most common allele to the other most common allele. You could try and phase out and compare haplotypes, or you could just look for strain cloud overlap. So maybe by specifically designing an algorithm for this purpose, you can come up with an accurate measure. And so this last metric of comparing strain cloud overlap is what we did for the strain level comparisons that are present within the program InStrain. And so I don't want to get into the technical details of how it's performed. If you're at all interested, we wrote up extensive documentation for InStrain. If you just Google InStrain documentation, you should be able to find it on the web. But essentially, from the ground up, we designed a new ANI metric called POP ANI, or population ANI, that essentially is able to handle microdiversity when it's doing these strain level comparisons. And this is necessary in order to use stringent thresholds like 99.9999% average nucleotide density. And so we've done a lot of benchmarks on this tool to kind of make sure it's doing what we think it's doing. I'm just going to show one benchmark here, but again, if you Google on the website, we've made all of the benchmarks clear. In this benchmark, which is an extremely simple benchmark, we purchased 3 tubes of the Zymo-Biomics Microbial Community Standard. This is a tube that just has 10 bacterial species in it at equal abundance that's used to validate metagenomic sequencing things. We purchased 3 of those tubes, did metagenomic sequencing on each tube, and then compared the genomes that we saw in these different tubes. And so ideally, we would see 100% average nucleotide identity between every comparison, because it's the same bacterial, it's the exact same strain that we sequenced each time. But what you see is that these other programs that are commonly used to do strain level comparisons don't report 100 average nucleotide identity, even if you sequence the same bacterial population twice and compare them. You're close, right? It's above 99.9% the same, but if 2 things that are actually identical are being reported as 99.9% the same, you'll never reach this 99.9999% threshold that we want. So it turns out that the reason why these other tools, why they cannot report essentially 100% ANI has to do with the fact that the comparisons they're doing don't take into account microdiversity. So again, when Zymo-Biomics grows up, E. coli in big vats to put into these tubes, population diversity is present. And if you don't microdiversity, and if you don't account for that when you're doing your strain level comparisons, for example, by using POP ANI, you end up thinking that these populations are very different, up to 3,000 years different, when in reality they're the same. And so this algorithm that we developed InStrain, in one scenario, it reported 2 organisms as having diverged for 2.2 years, which is incorrect. They haven't diverged for any years, but this is still within the accepted level of tolerance to say that, okay, InStrain is actually able to perform these very detailed strain level comparisons. So again, if you have any questions about this, I would encourage you to check out the online benchmarking that we have performed, because we spent a lot of time doing that. And so just to spend a couple minutes here talking about how I've applied this technique in my own work. Again, this is human microbiome stuff because that's what I do. We had this question about whether how strain sharing about strain sharing between mothers and infants. Sometimes this is called vertical strain transmission. This is somewhat well studied in infants from developed countries. There's a particularly famous study of mother-infant dyad pairs in Sweden that looked at how much of, when you're born as a baby, how much of the microbes do you inherit from your mom. And so in this example, we want to know actual, direct inheritance. And so it's an exact question where you really want to be stringent about, I want to know, did the baby get this B. fragilis strain from the mom, or did they get it from, somewhere else? And so applying these very strict thresholds, we kind of tried to address the same question, but in this case, this is looking at hunter-gatherers of Tanzania. So this is a one of the last remaining populations on Earth that practices a true hunter-gatherer lifestyle. And we found that, again, if you look at A, dyad pairs, so infants and their moms, share far more strains and a higher proportion of strains than non-dyad pairs. So that's infants and a different Hadza mom. But we also found, specific to the Hadza, that if you're a mom and if you're a non-dyad pair that lives in the same bush camp, you share far more strains than a non-dyad pair that lives in different bush camps. And so bush camps are kind of how the Hadza live their life. They're semi-nomadic people, and they generally live in bush camps of around 30 people for 3 months or so, and then they break camp and move on. But while they're living in a bush camp, they practice some forms of communal parenting. So, like, you know, some parents will go out to hunter and gather, or hunt and gather food, while some Hadza stay behind and watch all of the infants. And so during this communal parenting, we hypothesize that's when you're actually getting transmission of strains from people that are not your mom to these babies, which is very interesting. I won't get into B, but in C, this is arguably the most important finding, which is we compared what are the strains that are transferred in Hadza mom-infant pairs as compared to Swedish mom-infant pairs. And so you see at the phylum level, it's pretty similar. It's generally Bacteroides that are vertically transmitted in the Hadza and Swedish populations. But if you focus on specifically this concept of the Bacteroides and Prevotella and Vanished Blossom, you see there are stark differences between the types of strains that are transferred in Hadza and Swedish infants. I mean, I don't want to get into too many details here, but essentially, if you compare the adult microbiome of infants living industrialized lifestyles versus non-industrialized lifestyles, the two biggest kind of changes you see are that in non-industrialized people like the Hadza, you find Prevotella, which is a genus of bacteria, and in industrialized populations, you find Bacteroides instead, which is a related but different genus of bacteria. And so in Vanish and Blossom, it's essentially the same story. But so what this is showing is that the same taxa that are differentiating these populations of the Hadza and Swedish populations in adulthood are vertically transmitted between infants to moms. So it kind of shows that, for example, if you were to raise a Swedish infant like the Hadza raised their children, you still would not develop the Hadza microbiome, because you have to have inheritance from your mom of these strains, which are not present in the Swedish populations anymore. So it's kind of showing that vertical transmission may, potentiate these lifestyle differences. So again, I'm sorry to get so human microbiome on you, but I just wanted to talk a little bit about, the application of these types of strain sharing tools, which again can be used in any environment, but I'm most familiar with the human microbiome, so that's where my example came from. And so those are kind of the two biggest concepts I wanted to talk about with genome-resolved metagenomics. At this point, I want to touch on the other things I talked about in the introduction, but just give one slide each. One of them is the metagenomics of bacteriophages and eukaryotes. Typically, when people do metagenomic analyses, they're focusing on bacteria and archaea, but it's very possible to look at bacteriophages and eukaryotes as well. The tools for doing this are advancing all of the time. Some of my favorite tools right now are Beer Sorter 2 and CheckV for identifying bacteriophages, and UCREP for identifying eukaryotes. These tools all essentially work the same way, and that you develop a machine learning classifier to say, okay, of all of the sequences I have in my metagenome, which one of these are bacteriophages and eukaryotes, and which one of these are bacteriophages, and which ones of these are eukaryotes. And so you can run these tools somewhat easily and identify bacteriophages and eukaryotes in your sample. Again, in the human microbiome, what has this led to? With bacteriophage identification, it's led to this recent study that kind of identified new clades of bacteriophages with very large genomes that were unknown kind of prior to the study, again showing that the new life that you discover with metagenomics isn't just bacterial, it's also bacteriophage, viral. And so, these bacteriophages probably also resistant to culturing, which is why we haven't discovered them before, but you can discover these big clades with metagenomics. In terms of eukaryotes, I'm showing an example from a somewhat fun study I did during my PhD, where we sequenced the babies as well as the hospital room that the babies were born into. And in doing the sequencing of the hospital room, we also sequenced the sink where the nurses wash their hands And in the drain of the sink, we identified these two relatively unique eukaryotic genomes, one of which is a genome of a eukaryote that is somewhere in between a fly and a mosquito, and one of which is a worm that was somewhere in between a threadworm and C. elegans. And so, we don't think that our research nurse who's collecting these samples accidentally, sequenced an actual fly. We assume in both cases this is eggs of some sort that were sequenced that we got the DNA of. But it's just kind of very cool that when you do metagenomic sequencing, if you just look for eukaryotes, often you could find them and come up with relatively complete genomes of, again, forms of life that have not been sequenced before. In this case, it's almost certain that whatever genome we got of this insect is in a book somewhere, and someone knows about this organism and would like to have the genome that we assembled, but we are not able to know, what is the actual species of insect that we sequenced the genome of. We just have this genome that is relatively rare. So again, kind of a fun story. And I don't have any examples from it, but I also know that it's looking for eukaryotes in ocean metagenomes is very not common, but it has been done and is very cool. Specifically, again, with the program Anvio, there's been a lot of success there. So that's something that's very cool. Yes, functional consideration. This is a Twitter poll I put out a long time ago, about a year and a half now, asking why do metagenomics? And the most common response here was not, discovery of novel life or strain level comparisons, which is what I am a fan of. It was functional analysis. And so what we mean by that is that when you're doing metagenomic sequencing, as opposed to 16S sequencing especially, you're sequencing all of the genes on the genome, not just the marker gene. And so you can get an idea of what are the functions present in the metagenome, like acetate degradation, carbon fixation, whatever. And so unfortunately, I am not going to spend a lot of time on this topic because it is a vast topic with many different programs that can perform this, many different databases. And at least in my opinion, there's not really one pipeline that's the best. There's a lot of different ways of doing functional consideration. One is not necessarily better than the others. It's kind of the Wild West out there. If you have a protocol that works for you, or able to find a protocol that works for you, that's great. There's not really a lot of standardization in this field yet. But these are a couple of databases I'm listing here. But in all cases, functional consideration, functional analysis relies on the same basic concept, which is you assemble your genes, and so you have the amino acid sequence of your gene, and you compare it to a database where the database has amino acid sequences of a number of genes that have been characterized in the lab that have known functions associated with them, like a rubisco or something. And so you're essentially saying, okay, I have this gene, compare it to a database of characterized genes. If it's similar enough, I'm going to assume that my gene also performs that functions. So there's two kind of main caveats here. One is that these databases are incomplete, especially if you're working with a novel genome where it's a new form of life that really hasn't been seen before. Many of the genes on the genome will not be able to be annotated because they've never been characterized in a laboratory, so we have no idea what they do. And so there's really, no matter what you do, you can't really figure out what these genes do if they haven't been characterized in a laboratory. And the other aspect is it has the assumption that if these two genes look the same, they must have the same function, which is generally the case. But there are many examples where single point mutations in a gene or something like that will completely change the function, which is very hard to figure out computationally. So you have to kind of take all these analyses with a grain of salt because of these kind of two aspects. But they're very common, and there's a lot of tools to do them. And as revealed by this poll, it is kind of what people think of when they think of metagenomics, is this functional consideration. Great. And so the last thing I kind of want to talk about here before moving on to more practicalities type stuff is in situ replication rates. So when you sequence a metagenome, kind of for free, you get an idea of the growth rates of all of the bacteria that were present in your sample at the time of DNA extraction. And you can measure this based on something called PTR or IREP, et cetera. Depending, there's a lot of metrics to do this, but they all work on the same basic concept, which is relying on the fact that bacteria replicate their DNA from origin to terminus bidirectionally. So there's a single point on the chromosome where bacteria start replicating their genome. They replicate it, outwards, and it ends at the terminus. And this process generally takes about 40 minutes. And so for that 40 minutes, while the DNA is replicating, if you extract the DNA and sequence it, you will sequence more copies of DNA at the origin of replication as compared to the terminus of replication. And with careful laboratory studies, it's kind of been shown that by taking the ratio of DNA that is sequenced at the origin versus the terminus of replication, you can get a very accurate measure of doubling time, the doubling time of the whole population. You by looking at the peak to trough ratio, you can get a very accurate idea of the in situ replication rate at the time of DNA extraction. And so another program to do this is called IREP, which works on metagenomes where you don't know where the origin and terminus replication are. It's just based on the total spread of coverage across the genome. I will say also that IREP is calculated by InStrain as well. You get that for free if you run InStrain. But these growth rates are very, very cool, and you can kind of it's another aspect of the community, in addition to the abundance, that can be very interesting. And again, in the human microbiome world, it's been found that there are a number of organisms and diseases that are associated with differences in growth rates of organisms, but not abundance of organisms. So for example, sometimes, the abundance of Klebsiella pneumoniae is not associated with some disease, but the growth rates of Klebsiella pneumoniae could be associated with some disease. Which is, again, you get this information for free if you're doing metagenomics. So it's just another kind of very cool thing to do. And so for the last slide, before I move on to practicalities, I want to kind of give an overview again of all the things that I've talked about and kind of integrate them all into a single story, which is my PhD thesis, into how, kind of give an example of how you can combine all of these types of things towards some question that you have that you want to answer with metagenomics. So again, we talked about strain level comparisons, in situ growth rates, functional characterization, microdiversity, and then non-bacterial aspects of the microbiome, such as bacteriophages, eukaryotes, plasmids, and eukaryotic viruses. And so an example study where I've used all of these analyses is trying to look at the cause of the disease necrotizing enterocolitis. This is a disease that affects about 10% of very premature babies, and it's called necrotizing enterocolitis, and it essentially is where the baby's intestines rot. And so this is a X-ray photograph of a baby that's suffering from necrotizing enterocolitis. You can see there's all of these kind of gas vesicles going on in their stomachs. They get these very big, swollen, distended bellies. They're very, very sick. It has about a 50% mortality rate, maybe like a 30% mortality rate. So it's an extremely deadly disease. And for a long time, doctors and pediatricians thought that this disease was caused by, a microbial component. It's literally the guts rotting. It makes sense that this would be caused by a bacterial agent of disease, but no one had been able to discover what that cause of the disease is. And so a pediatric surgeon at the University of Pittsburgh reached out to our group, Jill Banfield, to say, okay, we know that not all bacteria can be cultured on plates. Maybe the cause of this disease is a bacteria that is resistant to culturing that we can discover with metagenomic sequencing. And so we recruited a very large cohort of babies, some of which that got necrotizing enterocolitis and some of which did not. And we kind of wanted to tell, is there any microbial component or any microbial components that are associated with the disease that we can discover with metagenomic analysis? So again, we kind of ran the whole gambit. This is an overview of the methods that we used, but as you can see, we did, genome-resolved metagenomics, which is where you do metagenomic sequencing. You assemble the short reads into long contigs, bin the contigs into genomes. Specifically, we looked at bacteria, eukaryotes, bacteriophages, plasmids, protein clusters, eukaryotic viral proteins, secondary metabolite clusters, virulence factors, previously implicated pathogens, protein families, IREPs, bacterial metabolism, sample diversity, clinical metadata, you name it, we looked for an association with this disease. And in the end, we discovered 4 things that are statistically associated with necrotizing enterocolitis development. IREP, secondary metabolite clusters, Klebsiella, and fimbriae. Just to talk really quickly, secondary metabolite clusters, these are groups of genes that work together to produce metabolites that are generally exported We identified those using anti-smash, which is just a program it's a different way of doing functional annotation. And fimbriae are they're typically thought of as a virulence factor that allow bacteria to stick to the intestines of things. They're very sticky appendages that bacteria produce to stick to things, which is generally not what you want in the human microbiome. And so what you're looking at here for each of these factors is the effect size of the difference between the babies that have necrotizing enterocolitis and the babies that don't have necrotizing enterocolitis relative to the days prior to diagnosis of necrotizing enterocolitis. And we did this because once a baby gets necrotizing enterocolitis, they're given tons and tons of antibiotics and all feeding is restricted, et cetera. And so the microbiome is very messed up by that, but we wanted to see prior to disease as the difference in the microbiome. And so the most striking thing you'll see related to IREP, which is this growth rate thing I was just talking about, is that immediately prior to necrotizing enterocolitis diagnosis, you see a huge spike in the overall growth rates of bacteria in general in the gut. And so there's definitely, this overwhelming growing that's happening of all bacteria in the gut prior to necrotizing enterocolitis development, which was unknown prior to the study. And again, kind of shows why genome-resolved metagenomics and artisanal metagenomics is very cool. You also see throughout the whole time prior to NEC diagnosis, you have an increased abundance of secondary metabolite clusters and Klebsiella. And also prior to diagnosis, you have an increased abundance of fimbriae. All of these, especially Klebsiella and fimbriae, are sensed by the human immune system, and the human immune system reacts to these things. Klebsiella is typically thought of as a pathogen, and so is fimbriae. And so if your gut sees these things, you develop an immune response against those to kind of try and attack them. And so all of this in another number of other studies that were published while we were doing this work and after we were doing this work have kind of led to the current conclusion that this disease, necrotizing enterocolitis, is not caused by a pathogen in the sense that we typically think of it. We think it is now caused by the infant immune system overreacting to pathogenic signals in general. So as I said, this disease only affects premature infants, not full-term infants. Premature infants have a increased abundance of this specific immune receptor called TLR4 receptors. And these TLR4 receptors react to things like LPS and Klebsiella and fimbriae. And so what we think is happening, or what the current state of the art thinking is that in premature babies which have an overactive immune system, if you get these types of things like Klebsiella and fimbriae, these bacteria don't cause the disease themselves, but the baby's overactive immune system reacting to these things leads to this feedback loop where you essentially end up with super amounts of inflammation here and also bacterial growth that kind of lead to necrotizing enterocolitis. And so again, I'm sorry to talk about human microbiome stuff so long, but I just wanted to crystallize all of these concepts into one study to kind of show how you can use them all, integrate them together. And I will just say that today, necrotizing enterocolitis, there's a lot more treatments for it, specifically related to human breast milk. It turns out if you give premature babies human breast milk, you have a lot lower chance of developing the disease. And we think this has to do with specific antibodies and other nutrients in the breast milk that tends to neutralize these types of bacteria. So wonderful. This is kind of the last that I have to talk about broad overview concepts and things like that. And I will now move on to talking more practicalities. And I'll also say if anyone has any questions or anything during this talk, please feel free to interrupt me. I'm more than happy to take questions as I go, but otherwise I'll just keep talking. So please interrupt me if you have any questions. Okay, wonderful. So when you're doing metagenomic analysis, there are kind of three different buckets of analyses that are possible, at least as far as I see it. There are Kamer-based analyses, gene-based analyses, and genome-based analysis. And again, there are hundreds of programs to do computational analysis, metagenomic analysis. And so I'm just trying to group these hundreds of programs into three main groups based on the core of how they work. So arguably the most popular types of methods today are Kamer-based analyses. This is like Kraken2, Bracken, things like that. And the main benefit of Kamer-based analysis is that it's very fast and computationally efficient. And so when you're doing a Kamer-based analysis, you're not emphasizing biology, you're more emphasizing speed and computational efficiency. And so the ways that these work is, metagenomic reads are typically about 150 base pairs long. Maybe you have 2 paired 150 base pair reads. In a k-mer-based analysis, you're only looking at 35 base pair sequences within your read. So essentially, a k-mer is a set length piece of DNA. Typically, it's 35 base pairs if you're talking about these types of k-mer analysis. And you essentially say, okay, well, here's my metagenomic read. Let's look at all the different 35 base pair sequences I have. Let's look for exact matches of these 35 base pair sequences or k-mers to what's called an LCA classification tree. And so you can say, okay, well, this read has this k-mer. This k-mer is also found in this species, and therefore I probably have this species in my sample. And so, again, the reason why k-mers are used is not for a biological reason. It's for a computational reason, which is that by looking like looking for direct overlap between these 35 base pair sequence in your sample and a database is computers know how to do that very well. So this is a very computationally efficient way of analyzing your data, which is great. You can run it on your laptop and things like that. But the downsides are that you're kind of removed from. It's not very intuitive to me in terms of the results that you get, and you can't really use it for gene level metrics. These are typically done for species level profiling. The other two techniques, which I'll talk about kind of together, are gene-based analysis and genome-based analysis. In this case, as opposed to k-mer-based analysis where you're chopping up your reads into 35 base pair sequences, in this case, you're aligning the entirety of the reads to either genes or genomes. And so if you're aligning to genes, right, you have resulting mapping where each gene has some reads aligned to it. If you're mapping to genomes, you have the resulting mapping as each genome has reads aligned to it. The benefit of aligning to genes is that you can, align to all genes that are known, right? You can map to the pangenome of each species that you know about to kind of exhaustively look for all genes present. Whereas if you're mapping to genomes, you're typically mapping to a representative genome from each species. So if the genome that you're mapping to of E. coli doesn't have some E. coli gene, when you're mapping to the genome, you won't find that gene. In terms of calculating abundance, if you're mapping to genes, you just essentially average the abundance of the genes of all of the genes in a genome to approximate the abundance of that genome. This is what Metaphlon 2 does and Metaphlon 3. If you're mapping to genomes, it's a very direct calculation of whatever percentage of reads are mapping to the genome is the relative abundance of the organism in my sample. And then if you're doing gene-based analysis, each gene is kind of analyzed in isolation of this is how much is present, et cetera. If you're mapping to a genome, you can kind of do this genes analyzed in context situation where you can see, okay, well, on this genome, I have reads mapping to this gene and this gene, but not this gene. So my population probably has this gene deletion. So that's kind of an overview. My preferred analysis technique and what we learned to do in the Banfield lab and what Jill Banfield kind of pioneered is this genome-based analysis. Some benefits of it are it is relatively computationally efficient, especially compared to gene-based analysis. Gene-based analysis can turn into very computationally expensive if you're mapping to large databases of genes. But I would argue the best aspect of genome-based analysis, genome-resolved metagenomics, which is what I'm talking about today, is the fact that it's very intuitive of all steps what you're doing. You're actually working with genomes and reads, and you kind of have a very intuitive sense for all of the steps of the pipeline and how your analysis is going, which sometimes when you're doing metagenomics, it's kind of easy just to get a result and not really know what's happening on the back end. When you're doing genome-resolved analysis, it's all intuitive sense and it makes sense. So that's what we'll be talking about today. That's what I like to do. And I'll also point out that for InStrain, we spent a long time writing documentation. There's, I believe, 100 pages of documentation on this website. So again, if you Google InStrain, read the docs, you will be able to find all of these figures and things like that. And so I don't know if I'll do this example quickly. I want to talk about breadth and detecting genomes in metagenomes. So if you map reads to a database of genomes, which is again what I will encourage you to do with genome-resolved metagenomics, you still have to say, based on this mapping, which genomes are actually present in my metagenome or not. And so you have to have some sort of threshold of, okay, well, if one read maps to my genome, I probably don't have very much confidence that the genome is actually present. because, maybe this is just a read that is, truly belong to another population, but it's just mismapped here. Maybe it's a transposon, et cetera. And so you need some sort of threshold to determine if a genome is present or not. And so the two metrics that are typically used are coverage and breadth. So coverage depth, or sometimes just referred to as coverage, is the average sequencing depth at any position along the genome. So right, the depth at this position would be 10 because there are 10 reads mapping there. The depth at this position would be 1 because there's 1 read mapping here. And the coverage depth across an entire genome would be the average of all of that. So in this case, it would probably be like 3 or something like that, 3x. Whereas breadth is the percentage of the genome that is covered by at least 1 read. So in this case, you don't care how many reads are mapping to this part of the genome. You just care that it has at least 1 read mapping. And so, this section is covered by a read. This section has no reads. This section has no reads anyway. So the breadth of this sequence would probably be about 0.8 because about 80% of the genome is covered by at least 1 read. And so this question, which again I posted on Twitter, is a poll, which would give you a greater confidence that the genome is present. If you have 10x breadth, so you have 10% of the genome covered by reads and 1x coverage, or 0.1x coverage. So maybe I'll pause for 5 seconds so everyone can kind of, in your head, think about what you would answer. And then to help you along, here is a kind of diagram that I drew of what's going on here. So in both cases, you have 10% of the genome covered by reads, so 10% breadth. In this case, with 1x coverage, or no, 0.1x coverage, you have an average of 1x coverage across the genome, which means you have different reads mapping. You have 1x coverage for 10 kb, which means overall you have 0.1x coverage. Whereas in the scenario we have 1x coverage and 10% breadth, you have 10x coverage for just 10% of the genome. And so, yes, in terms of the Twitter poll, most people answered 1x coverage at 10x breadth, which is this scenario. But I would argue that this scenario gives me far more confidence that the genome is actually present in the metagenome. And what this has to do is what this example is trying to illustrate is the concept of mismapped reads. So in this scenario where you have a mapping of lots of reads mapping to a very small percentage of the genome, probably how I would explain what's going on here is that you have a transposon or a bacteriophage or some mobile element in your sample that is also present in this genome, but the genome isn't actually present in your sample. So you get lots and lots of reads mapping to one portion of the genome, but you don't have any reads mapping to the majority of the genome. And so in this case, if you're just talking about, if you're calling an organism present just because it has 1x coverage or it has at least 100 reads mapping or something like that, you're going to count this genome as being present when likely this genome is not actually present. It's just one portion of the genome that is present. Whereas in this scenario, again, you only have 10% of the genome present, so maybe you don't want to call it there, but this is far more indicative of the genome is there, but it's just not a high enough abundance to get this whole coverage. Like you're having reads map randomly across the genome, suggesting that the genome is actually there, but yeah. And so you can kind of estimate, I don't want to get into the details here again. I'm sorry I keep referencing this website, but I really get into it on this website. You can, by looking at breadth and coverage, you can come up with sorts of rational thresholds for doing genome detection in metagenomes that again are rational and make sense and are kind of based on statistics. So if you, in this example on the left here, we just took a bacterial genome, simulated reads from it, and backed reads back to it to say, okay, well, what is the relationship between coverage and breadth in the case where all of the reads are coming from the genome of interest? And what you see is that at about 1x coverage, you should have about 50% breadth. So that means if you have 1x coverage, you should have about half of the entire genome covered with reads if you just map reads randomly. And by the time you get to 5 or 6x coverage, you should have full breadth, 100% breadth. All of the genome should be covered by reads, and you can kind of map a function here. In strain, you can kind of measure this data and get an idea of is the breadth that I have in line with the breadth that would be expected given my coverage value. And the math here gets a little bit confusing, so I don't want to get into the details, but essentially the point of this what I'm emphasizing and why I brought this all up is that determining or coming up with thresholds for determining if an organism is present or not in your metagenome is something you have to take kind of seriously and is important and will impact your analysis. And there are rational ways of doing that, specifically by looking at breadth and coverage, which is what I like to do. The next kind of practical aspect here is you have to decide when you're doing genome-resolved metagenomics, you have to decide what you want to map your genomes, or what database of genomes you want to map your reads to. So there's kind of three options here. One is you can just use an existing genome database. So here's some examples of genome databases you can just Google right now. Download all of the genomes, map your reads to them, run InStrain or whatever tool you like, and get an idea of the presence of all of these genomes in your sample. Alternatively, you can generate genomes de novo. So this is de novo genome assembly. In this case, you have your metagenomes. You assemble genomes from those reads. So you get de novo genomes that you created from your metagenomes yourself. You dereplicate them, which I'll talk about on the next slide. Then you map reads to them. So and an alternative would be to combine your de novo genomes with a public database into a combined genome database, which is what I argue is the best scenario. So the benefit of A is that all bacteria that we know to exist that are in these databases, you can detect if you use one of these genome databases. But especially if you're working with somewhat novel environments, it's very common to have discover new genomes in your metagenome. And so if you generate genomes de novo, in a case where your sample has some new form of life that isn't in a database, new bacterial species, you can map to it and you can detect it, right? If there's a new species that isn't in this database and you map to the database, it's just going to be invisible to your analysis. But assembling genomes is a non-perfect process. And just because you don't assemble a genome for an organism doesn't mean that genome is not present in your sample. It's very possible to know that E. coli is in your sample, but in this particular assembly, for whatever reason, you don't recover an E. coli genome. And so in this case, if you're just mapping to your database of genomes you assembled de novo, you're going to, all genomes that you're not able to disassemble for whatever reason are going to be invisible. But by doing a merging of your de novo assembly in a public database, you kind of get the best of both worlds, which is where, in the case where you don't assemble a genome, but it's known, you see it in a public database genome. And in the case where the genome is novel and not present in a public database, you're still able to see it in your mapping database at the end that you map to. It's combined genome database. So I hope this makes sense. And so kind of the last topic I want to talk about is the dereplication step, which is when you have a genome database that you're mapping to, it's very important that you don't have two genomes that are very similar to each other in your database. And for the reason for that is that when you do metagenomic mappings, typically, if a read maps equally well to two different genomes, the mapping algorithm will just randomly assign it to one or the other, which for a number of reasons is typically what you want it to do. This is a good, this is what they should do, but it causes a problem where if you have two genomes that are near identical to each other in your database and you map reads to them, many reads will map equally well to those two genomes. And so rather than seeing one genome at 5x coverage, you'll see two genomes at 2.5x coverage. And this is kind of illustrated here, where we did what I just said don't do, which is map to a database where it's just lots and lots of genomes, no dereplication has happened. You see the scenario where the orange dot is the actual genome of the organism that's present in this sample, because this is a benchmark, so we know that is the case. You see that you have lots of reads mapping to the genome that should be there, but you also have lots of reads mapping to genomes that are related to this genome in terms of average nucleotide identity. So this genome is 99.8% the same as this genome. So you're detecting a lot of reads mapping to that genome as well as further out accidentally. And so essentially what's happening here is in all cases where you're getting a read map to this genome, it's a read that maps equally well to the orange genome and this blue genome, and the algorithm is doing what it's supposed to do, which is randomly giving it to this other genome down here. And so if you look at the same mapping file, but you just remove all reads that are multi-mapped, so they map equally well to 2 positions, you don't see this phenomenon anymore. You just see the genome detected that you should detect. For a number of reasons, you generally don't want to remove reads that map equally well to 2 places, because you end up missing a lot of the genome's interesting things like bacteriophages and mobile elements, and you get far less reads mapped, so you can see the y-axis is very different in these 2 scenarios. So rather than just removing multi-mapped reads to handle this problem, it's best just to dereplicate your database prior to mapping reads so that you don't have any genomes that are, for example, 98% the same to each other, and kind of remove multi-mapped reads that way. And so in coming up with a threshold for how similar can 2 genomes be before I'll let them be in the same database, right? Like at what level should we perform this deep replication? You can do it, as this graph would suggest, at up to 98% average nucleotide identity, but I prefer to do it at 95% average nucleotide identity, which is the threshold of bacterial species. I don't want to get into this too much, but I kind of love the topic of bacterial speciation. And this is a paper showing that at 95% ANI, if you just look at how related are 2 genomes in these different environments with metagenome assembled genomes, no biases, whatever, you see that there is these groups of organisms that are 95% average nucleotide identity to one another, and then a break, and then these other groups of organisms that are not the same species. And this break here of showing that there is kind of the concept of species is real and makes sense, and the differentiation is at 95% ANI, makes me, in all of my analyses, typically use 95% average nucleotide identity when you're dereplicating your database, which is good because it removes the possibility of multi-mapped reads, but it also means that your ultimate metagenomic analysis is very simple from a simple and intuitive, in that each bacterial species has 1 genome in your database. So you can track the abundance of bacterial species based on your 1 representative bacterial genome in your database. And so again, if you're interested in performing this on a practical level, there's a lot more information in the important concepts page. And then the very last thing I want to talk about, and I only want to talk about it very briefly, is kind of on a practical level, right, at your computer, what does running InStrain look like? And I should again emphasize that this is the program that I wrote, so of course it's the one I like to talk about. It's the one that I use. I think it's very good for specifically doing genome-resolved metagenomic analyses. But there are number of other programs out there that perform the same thing. There's nothing particularly special about InStrain other than that it can measure these microdiversity things that we specifically programmed it to do. But Ambio is another very popular tool for doing stuff like this. InStrain, though, in particular, the way that it works is that you have your microbiome samples, you generate metagenomic reads from them. So this is, you extract DNA, do metagenomic sequencing. These files are typically called FASTQ files. You then map your reads to a database of genomes. We just talked about how you create that database to create a mapping file, which is typically called a BAM file or a SAM file. And so you have your reads now aligned to your genome database. You then run this program InStrain Profile, which generates a folder from each of these mapping files that has information about the microdiversity of every genome, the coverage, the breadth, lots and lots and lots of information is generated here. And then if you want to compare them to do strain level comparisons, for example, this PopANI metric I was talking about, you run another command where the input is these InStrain profile folders in order to generate an InStrain compare object, which has information about the PopANI between all of these different genomes and things like that. And so, and again, one last shout out. We tried to pick a lot of documentation for this. If you Google InStrain documentation, you'll find it. And I'm also very available to questions online or email if anyone has any questions running on any of this stuff. I'm very happy to help. And so with that, I'll just briefly acknowledge my current lab that I'm in, the Sonnenberg lab. This is a photo of us a few weeks ago, and this is when I still had long hair. Justin is a great advisor. I very much love being in his lab right now. And Jill Banfield, this is when I got my graduated in 2019, got my PhD in her lab. Instilled this love of metagenomics in me that I still have very obviously. So I have to thank her as well. And I'm now happy to take any questions that anyone has.
Oh, amazing, Matt. Thank you very much. I think we do have not a few actually, some questions at the chat.
Or
We're gonna give it to you just a
can I
second.
ask a first one since I'm in the chair? Just a moment, Julian. Matt, I have a analogy, and maybe you can agree or disagree and see if works. When you talk about specific about breadth versus coverage, okay? And you can tell if my analogy is right because you have so many undergrads all over the place watching this right now. And would an analogy be like a DNA sequence, okay, technology, be a bullet or a bullet from a machine gun that you fire at the target, and you know it'll be more interesting to hit the target in several different places instead of hitting bullet after bullet the same region of that target when you compare breadth versus coverage. So I would agree with you that the more bullets spread along the target, would be a better idea if that the genome is present or the target is present or not in a bacterial or microbial community. Is that right?
Yeah, totally. That's exactly right. And yeah, it's like I can't improve. Yes, you said it great. Yeah, coverage. If you have 100x coverage, it'd be like the bullet, going 100 times in the same hole in the target. Whereas if you have 100x breadth or 100% breadth, it would mean every single portion of the target is now gone because you have, because it's been hit.
Great. Juliana, do you have questions from the audience?
Yes, we have some questions from the chat. I think it's great. So Larissa is congratulating you, and she has a question about what do you think we have to be most careful with when working with metagenomics bioinformatics?
This is a super great question, and I have a lot of different answers come to mind. I would say counterintuitively, I think some of what I see a lot of kind of newer people to bioinformatics struggle with is that there's so much to know, and there's so much knowledge out there, and there are so many programs. A lot of people are intimidated that they want to be doing the best analysis and that they want to make sure that their methods are like the most robust. But there's not a lot of standardization. Like I would say, don't be too scared about getting your analysis out there, and don't let perfect be the enemy of good. There's just so many different things to try, and you kind of have to recognize that you'll never be able to compare, your same analysis using every different program. And so it's don't get too stressed out about being perfect, I guess would be my my primary piece of advice. And then the other thing in terms of like more, what is actually to work out for. When looking at, I spent a lot of time talking about these tools that are not genome-resolved metagenomics. Tools that are based only on databases and you have to be worried about database completeness. It's very easy to miss things that you're not seeing, to not see things because they're not in your database, which is not necessarily a problem if you're aware of that. But the other thing is that I tend to be wary about things that are so algorithms and programs that are so complex that you can't really understand what they're doing. There's a lot of like, I guess it's just because I'm not a statistician, but oftentimes there's these programs where I'm trying to read the paper and it's very confusing and I don't understand what's going on. And I worry about those. And I prefer analyses that are more intuitive and straightforward and you can kind of understand what's going on. But that's at least my 2 cents.
So, and a follow-up question that you asked previous speaker, Matt, is for this is a problem, right? So, especially for students, right? So they run different workflows to answer a biological question, a specific biological question, and the output is a little bit different for each workflow. So you have a gene present in one, and the gene doesn't appear in the next one, and so on. So the question would be: is my pathway present or absent, and is dependent on what kind of workflow I'm using? So what would be your thoughts about that when you have this? I'm not saying about something that's completely different, because then you will have a red flag right away, right? So it's something funny going in. But you have small, very small difference, but they are important for your description of that microbial community, or what's going on in terms of function, for instance.
Yeah, totally. It's a very common thing to have happen. It's very stressful. It happens to me all the time, and I think it's just inevitable in the fact that when we're doing metagenomic sequencing, we're kind of just trying to take our best guess at what's actually going on. Like I like this analogy I saw on Twitter the other day, that like metagenomics is like trying to look through like a couple layers of like frosted glass, trying to see what's on the other end. And so like, it makes sense that different algorithms will make sense of the underlying biological data in slightly different ways. And I don't have a lot of advice about how to deal with it, other than the fact that it shouldn't stress you out, and it's just a reality of doing this metagenomic analysis. If you report different things in your paper about, okay, well, it's I guess all algorithms are imperfect. None of them are going to give you an actual perfect representation of what's going on. So of course, different algorithms will have different interpretations of what is going on. And metagenomics is very powerful, and it lets us see things we've never seen before, like discover new life and things like that. But it's far from perfect, and it's still an approximation of what is going on. And so it's very important then to not get too hung up on like any particular one result or algorithm or p-value or something like that, and kind of try and more look for broad patterns and things like that. But in the specific example where there's one gene that you really care about for your analysis, and you get 5 different answers from 5 different programs, reporting the 5 different programs results and interpreting however you want is appropriate and fine. And I would encourage people not to be too worried about the details on that level. Does that make sense?
Yeah, definitely. Thank you. Georgios, we have the next one.
Okay, I guess we can go to, okay. So Jessica is asking how to reconstruct genomes, considering that most sequences are from conserved regions that could be presented in many different bacteria.
Yeah, this is a wonderful question, and extremely complex question. This is totally legit. So without getting into too much technical details, when you perform genome assembly, when you go from short reads to full genomes, which is what this question is asking about, this is done using a De Bruijn graph. And I don't want to get into too many details, but essentially, if in your reads you have 2 genomes that are very similar to one another, like 99.9% the same to one another, when you do your metagenomic assembly, the assembler will combine them into one genome that is a hybrid of these 2 other genomes, which is great. If you have 2 organisms that are very different from each other, say 99.5% ANI from one another, the assembler will just create 2 separate genomes. And the De Bruijn graph is able to handle this, and it's not a problem. When 2 genomes are somewhat close to another, but not exactly close to another, say 98% identity, I believe is like the danger zone, 97 to 98% identity, the assembler will completely struggle, and essentially you will end up with no assembly from either genome. You'll get this very fragmented hybrid situation where the assembler is not able to differentiate these 2 genomes or able to combine them into one sequence. And so that's one aspect of this. And that's when I said that just because you don't assemble a genome doesn't mean it's not there, right? If you have the same genome at the same abundance in 2 samples, but in one sample there's also a related genome, you won't get a genome in the sample that has the other genome, but you will get a genome in the sample where the genome's alone. So, metagenomic assembly is very complicated, and just another one of these many aspects that make genome-resolved metagenomics difficult. So that's one thing. But this question in particular is talking about conserved regions, and so depending on how long your reads are, and depending on the algorithm used, and depending how conserved it is, you may end up, the assembler may be able to incorporate this conserved region into your genome. Or if it's actually identical and it can't, the assembler will just break at that conserved region and you'll lose it. So for example, if there's a transposon that's present in 2 genomes, when you assemble your genomes, you will just end up with a break, like that sequence will be broken, and you'll have two separate genomes or two separate scaffolds in the end that don't have that conserved sequence. And so if it's a perfectly conserved sequence, you kind of can't assemble it. It's in two samples. The assembler will break. But if you, for example, assemble, if you map to a genome that has the sequence, it will be there. But I will say, okay, this is true. This is something that happens, and it's something to worry about. But in reality, like from a practical perspective, this doesn't happen that often. It's really only transposons and phages that are in multiple genomes that are that conserved enough that it will break the assembler. Transposons are the big one. And so you're typically not going to miss out on important functions due to this, because it's generally transposons that are doing it. But it is something to be aware of. I talked a little bit about that, but yeah, great question.
Sounds great. Actually, I have a follow-up question on that. So do you usually use, can you combine different long read and short read sequencing technologies to, make a way out of this problem?
Yes, totally. And that's a very promising area going forward is these long read assemblies with like nanopore or PacBio that explicitly can span these gaps. And if you have this identical region that is breaking your assemblies, but you have a long read that can span, the transposon, that can fix it that way. The long read technology is very exciting and very new. It hasn't really been fully incorporated into a lot of metagenomics workflows yet. It's kind of like more of a specialty thing. And I think a lot of the reason for that is that right now, if you're doing metagenomics, typically you're sequencing very deep, and it's very expensive to sequence very deep with these long reads. It typically works better for like isolate sequencing or for relatively shallow metagenomes. But if you are lucky enough to have long reads, you can definitely do these hybrid assembly programs that do leverage the long reads specifically for this function. Yeah, it's a great point. And hopefully in 10 years, it'll be very common and we won't have this problem anymore.
Yes, I agree. Okay, we have more questions. Let's just wait for them to pop up. Okay, so Taina is asking if we can access bacterial evolution using metagenomics, for example, based on mutation rates or mutation sites.
Yes, totally. This is a fantastic question and something I'm very interested in, and just from a curiosity perspective. So specifically, I'll answer this question primarily from the perspective of looking at evolution. There's kind of two aspects to this. First is looking for evolution within a sample, right? If I talked a lot about microdiversity. If you have microdiversity in a sample, it can be fun to try and reconstruct how that sample evolved based on mutation rates and sites and things like that from the metagenome that you have from the sample. Unfortunately, this becomes somewhat difficult due to the fact that you have only short reads. So if you have a variant on one site of the genome and a variant on another site of the genome, because of short reads, you can't really know if those are on the same piece of DNA or not. So this type of analysis on one sample is typically easier to do if you have long reads or if you have isolate genomes. But you can certainly start to get at this types of evolution questions with microdiversity, specifically with genes and things like that. And if you're interested in doing this in your own work, I would encourage you to look at, I keep plugging it, but the interesting documentation, we kind of go into how to do this with genes and stuff. The other aspect to this question is looking at evolution across different samples, in which case you kind of can just use metagenomics to reconstruct, a single consensus genome from each sample and compare them in that way. And in that type of analysis is you kind of get linkage because you have the different. Yes, that's perfectly valid to do and very exciting to do, and I love it. And the answer is yes.
Great. I also have here with me a question from Christiane Thompson, Professor Christiane Thompson. She's asking if, in relation to speciation, what are your thoughts on Phlorococcus collective? A single species Phlorococcus marinus instead of several genera of Phlorococcus. I'm not sure if this was clear. I do know the question from WhatsApp.
Yes, I definitely understand the question, but I unfortunately don't know. I'm not in the world of Prochlorococcus and ocean metagenomes, and with speciation in particular, it is a passion of mine, and I very much appreciate the question, and I like thinking about things like this. I'm not an expert, so I don't want to give my 2 cents, but I will just say that in terms of speciation, I like this 95% average nucleotide identity threshold to define species, and there's other work on kind of showing this, but I don't think it is universally applicable. There are certainly going to be weird scenarios where, maybe Prochlorococcus or these other genomes that are, species that are in the ocean. I would not be surprised at all if the paradigm that I like of this 95% ANI cluster is broken, for example, in these unique circumstances. And, I think that this speciation question is applicable, in 95% of cases or something like that. But special cases like that, I know that population genomicists have published on it and really looked at these different population structures, and different structures certainly exist. And I would not be surprised if that was the case. I don't know, so I don't want to speculate.
It's great. Thank you very much, Matt. I don't know if we have any further questions.
I'll have a couple more.
Oh, yes, we do have here. So do you think the use of artificial intelligence can make can help metagenomic analysis?
Another great question. I love all of these questions. Artificial intelligence, machine learning, advanced statistics, it's kind of like this gradient of just how much do you want to let the computer take over and kind of figure things out for you, without they call it black boxing it, not really understand what the computer is doing, but letting the computer kind of figure stuff out itself. I've done a little bit of this work myself, looking at specifically machine learning and trying to train machine learning algorithms to find things. I talked a little bit about this example of necrotizing enterocolitis. We tried to design some machine learning algorithms to predict it that had moderate success. I think a couple of things. One, this is kind of some people are doing this already, applying artificial intelligence and these kind of deep learning approaches to metagenomics. And there are some scenarios where it seems to be very useful and helpful. Google just recently developed this thing to predict protein folding, which is helping metagenomics a lot. A lot of these programs we talk about that differentiate eukaryotic and viral sequences are based on machine learning or artificial intelligence approaches to say, okay, well, I have the sequence. Is this actually a bacteriophage, a eukaryote, a bacteria? So for some scenarios, it's very helpful, but it kind of breaks down when you have very specific questions, just because of, as we've talked about, metagenomics is an imperfect representation of what is actually going on. So oftentimes these algorithms will kind of pick up on, little differences that you wouldn't expect. So for example, if you used a couple of different sequencing centers in your study, the artificial algorithm might, it ends up picking up on cofactors and I would say there's definitely a use for it, but it has limitations currently because the data that you're feeding these algorithms is imperfect. And oftentimes it requires kind of like a human brain and a biologist's brain to figure out, okay, well, what are the aspects of this study that I can trust more than other aspects, and what aspects should I focus on in trying to answer my question? Yeah, I hope that answers the question.
I'm sure it does.
I have another one. What would be the ecological importance of this microdiversity that you find in specific groups? You are talking about, and if you exclude pathogens from this process, okay, is this about resilience of that specific group, or adaptations, or preparation for environmental cues that you need these micro variations or micro diversity?
Yeah, it's a perfect question. And I wish I knew the answer. I mean, this is a little personal, but I wrote a grant trying to propose this exact question that didn't get funded. But I think that so a couple of things. Microdiversity is it's just going to happen all the time, no matter what, just because of how DNA polymerase works and how bacteria work. So there's not always a reason for it, but because of the way that evolution works, it always has certain characteristics, like, a deleterious mutation in your RNA gene or whatever, that population is going to die. So just by looking at microdiversity, even if it doesn't have an ecological consequence, you can get an idea of the evolutionary pressures that are faced by this microbial population. So for example, we've done studies looking at, again, the Hadza hunter-gatherer microbiome, and you see, a lot of microdiversity around genes that have to do with interactions with the immune system. You see microdiversity around genes that have to do with inflammation, and I think we're in the early days of really understanding it. But in terms of consequences, I would love to know if it helps population resilience. I don't think that we really know yet. I think it's hypothesized that it does. Certainly, if you look at like Shannon diversity between different species, the more diversity you have between species, you think of having a more resilient ecosystem. I would hypothesize along those same lines that having more microdiversity would result in a more resilient species population. You have more sample space or more sequence variation that you can call upon if there is a changing environment. But at least I'm not aware of publications kind of showing this directly.
Okay. Do you have any more questions, Ivan?
I guess that's it.
And so I will make
fantastic questions.
I have another last one, so you can conclude, Matt. Very general question about the vertical transmission that show up very clearly on the human gut. So I have several questions, and they pertain to the same point, right? So can you reassemble, or what's the current data about reassembling the human microbiome by introducing like probiotics and different artificial communities, specific diseases, because this correlates also to any organism, right? Even sponges on the ocean and coral reefs that suffer from different diseases and so on. So how your thoughts about this force reassemble of the microbiome?
Yeah, it's a super great question, and a very intense focus of research right now. I mean, the human microbiome is very big. Where I am in San Francisco, it's there's a bajillion biotech companies that are trying to figure this out right now. In general, kind of as you say, human microbiome assembly is similar to other microbial communities in that it's very structured. Like you have like these, you think of foundational members that then lead to other members and then lead to other members. And it's generally thought of as this progression from a simple community to a more complex one that kind of proceeds in these organized steps of like, first you get these, when an infant is born, their gut is essentially sterile. And it also there's micro oxygen levels in there. So you start with like facultative anaerobes, and then you move to an anaerobic environment. Anyways, so there is this progression there. One point I'll make is that we kind of understand this process much more in industrialized infants than we do in infants living non-industrialized lifestyles. So I think it's very important to study people outside of these wealthy industrialized nations to actually understand how this process happens outside of America and Europe. But in terms of introducing probiotics and things like that, it's confusing. It's there's a lot of studies, specifically the probiotic that people think about is Bifidobacterium infantis, which is a bacteria that has been correlated with having, if you have this bacteria in early life, you're less likely to have some sort of immune and allergies and asthma problems. But just because you ingest a microbe doesn't mean it can live in your specific microbiome, that has to have a niche open to it. And so oftentimes there's studies that will give this B. infantis probiotic, and you'll kind of find it in the gut, but it'll be at 100 to 1,000-fold less abundant than bacteria or than other bacteria or like other infants that naturally acquire this B. infantis. And the other kind of aspect to this is that because the human microbiome is so important, human breast milk has a lot of structuring role on the human microbiome. So for example, not only does breast milk contain nutrients for the baby, just to like feed it, but it also has a lot of what are called HMOs, which are components that the human body cannot digest, but the bacteria in your gut can digest. And so B. infantis, for example, really feeds on these HMOs. So if you're breastfeeding your baby, that it's thought that, so the breast milk not only contains specific food for the microbes that you generally want to have as a baby, but it also contains a lot of IgA, which is an antibody that specifically binds members of the microbiome that you don't want there. And so essentially, there's not a lot of substitute that we know of yet for breast milk in terms of its structuring component of the microbiome, and having breast milk in addition to some of these probiotics does seem to be helpful. But I will say that sometimes people get stressed. So that's my answer to the question is the jury's still out. It's a very open question of debate. And I do think that it's important for human health. And you know, probably in 20 years, we'll know a lot more about how to direct microbiome evolution for health. But I will say sometimes people get stressed out about, you know, oh, I fed my kid, my baby got formula feeding, or my baby was delivered by cesarean section. And so you're worried about its health. But I just want to point out that at least within industrialized populations, the effect size that we're looking at is not very high. It's not like, oh, okay, I was born by C-section, I'm definitely going to have all these allergies and asthma, or I was fed formula, I'm definitely going to have allergies and asthma. You know, it's like 2% differences or something like that between the groups. So it's not extreme, but it's definitely present. And so trying to understand, yeah, that's just all I want to say.
Great. Well, Matt, I think that's all the questions we have for now, and we are out of time. So I really thank you again for the very nice talk. Really nice to have you here, and really appreciate your help with our workshop.
Yeah, thanks again for having me. This was super fun, and I very much appreciate all these questions on here. It was super great questions. This was anytime. It was great seeing you again.
Okay, great. Thank you so much. Thanks so much.
Thank you so much, Matt. It was a pleasure.
Bye, everyone.
Bye-bye.
Thank you very much.
So we are done for today. We have an amazing lineup of speakers, and tomorrow will be the same. So we will meet here on the same channel, the same time, for the next round. I hope everybody have appreciated the very important and nice talks we got this morning. Thanks so much. See you everybody tomorrow. Bye.
Bye-bye. See you tomorrow.